And good morning everybody, and welcome to the back deck of the RV Falkor as we watch the initial descent of ROV SuBastian. This is dive number 388. This is a continuation of the voyage where we're exploring the Seamounts, canyons and coral reefs of the Coral Sea. Today we're at towards the northern end of the Great Barrier Reef, sitting offshore from Ribbon Reef 9 and 10, and we'll be taking a full day dive right from the sea surface today. We'll start off with a midwater plankton dive, photographing slowly through a timed descent the jellyfish and other planktonic life before we hit the seabed at 1500 metres later on in the afternoon. My name is Nichol from Geoscience Australia. Today we have Dr Dhugal Lyndsay from JAMSTEC in Japan joining us live through YouTube and Facebook to provide commentary and direct us to locations where we might sample some of the unique jellyfish in particular that Dhugal's able to see and identify as we move through the water column. We're currently at a water depth of a bit over 100 metres, so still within those warm surface waters. Good morning, Dhugal.
Good morning. Thank you very much for having me here. Oh, we've got a jellyfish already, it seems. Just coming up in the middle of the camera here. We'll see whether it's a jellyfish or just one of the other gelatinous animals that are very common in the deep ocean, especially in areas with not so much upwelling and many nutrients, such as this. Wow, that's weird. What have we got? Looks like it's 3 salps, perhaps. Salps are tunicates, kind of a sea squirt that swims around in the water, sucking water in one end of their body, filtering out the food and pushing the water out the other end to help them to swim. These guys look to be attached around the mid-line of their body. Wow. And there looks to be a baby also attached to one of them. We'll have to get out the books to work out what this is later. If we can get a shot of them from the side, we're currently looking at them like down into the body. Salps, you can use the number and shape of the muscle bands that are on each side of the barrel that allow the animal to pulse and bring in water and eject it again. The number of them and whether they're fused to each other or single is very useful for the taxonomy of salps. Although they look like jellyfish, they actually have a notochord, which is like a backbone, like the nerve that runs through your backbone. So they're actually closer to fish taxonomically than they are to jellyfish. And so we have 3 large mature animals and then one clone inside one of those zooids. Okay, we've got a variety of angles there. That should let us tell what they are. Thank you very much. Let's keep on going down deeper and see what else we can find. Salps have a sexual stage and an asexual stage, one of which gives rise to clones of themselves, and they form huge chains. And the shape of the chains, how they're attached to each other, helps you tell what genus of salps there are. For example, the genus Helicosalpa, all of the salps are attached to each other in a helix. There's Cyclosalpa when they're in a circle. Salpa, they're attached end on to each other, etc. So just by looking at the shape of zooids and how they're attached to each other, we can pretty much tell what the genus is. I wasn't really familiar with that last one, so I'll have to crack open some books to be able to identify that. My speciality is more in the cnidarians, which are the jellies that have stinging cells, and also the comb jellies, the ctenophores, which, although they are transparent and gelatinous, are not very closely related to the true jellyfish at all, really. They're in a different phylum. And rather than stinging cells, they have cells that adhere or stick their prey to their tentacles to allow them to eat. And they have 8 rows of little combs that they move through the water like paddles to allow them to swim. We'll probably be seeing a range of different comb jellies, as well as regular jellyfish, and all sorts of other gelatinous plankton during this dive.
Dhugal?
Marine snow that's, yes.
Yeah, Rob here. Yeah, yesterday in one of our deeper dives, we saw a really distinct and quite large ctenophore. It had lots of red on it, so it was quite deep down, but it had had the distinctive 2 trailing tentacles. It was near the seafloor, but really quite a large red ctenophore. But you could see the bristles, the comb-like bristles on the side as it was swimming.
Okay, was it long and oblong, like this kind of width to length ratio?
It wasn't really long. No, it was more globular. It was kind of a bulky-looking ctenophore. They were very distinct red colour.
It sounds like it's undescribed, an undescribed species. At present, the only red cediped, which is the guys with the 2 tentacles that's described, is Vampyrotina off the coast of the United States. But that's long and oblong. So you might have something new there. That'll be great.
So Dhugal, one of the questions which is coming up quite frequently from emails and chat and so forth is about the colours of these organisms and why are they red and purple and all those wondrous colours, given they're sitting down in the pitch black for the most. Any ideas there?
Yeah, so up at the depths we are now, where there's still daylight penetrating down, well, maybe we wouldn't be able to see it, but the very sensitive eyes of the midwater animals can see it. At these depths, the jellies are mostly transparent and they're mostly small because they're trying not to stand out against the light. To make your body transparent, it actually takes a lot of energy, because when you eat something, you have to digest the pigments until the pigments are gone and broken down, and keep your body transparent, which takes energy. So as you go further down, where there's less and less light, the animals are not forced to use that energy so much. Is that a fish? An eel? Leptocephalus larva, perhaps?
Yeah, it has a really distinct eel. Yeah.
Nice. You can see his head.
So what kind of fish? What kind of fish there, Dhugal?
An eel. It's an eel larva. If you look at the head, we might be able to tell if it's a midwater eel or if it's like a moray or something else that may have an eel larva like this. You need to see the head, and usually you need to count the number of muscle bands along the body, which is going to be very difficult because they're transparent and the background is also slightly lit up from the ambient light. That's cool. Now we are possibly in the thermocline now. Eel larvae are known to eat appendicularian houses, larvation houses, sometimes even jellyfish, and that kind of food tends to congregate at the thermocline, which is where the temperature suddenly changes from warm to cold over a very short vertical distance.
We're still within the surface water. Yeah, I wouldn't say we're still a little bit shallow for that, but we are in this surface mixed water layer. So, it's within wave base, and it's still warm. It's 20.9 degrees C where the ROV is at the moment. Okay. So I would expect the thermocline's a little bit deeper down.
Okay. So there's enough food here for them to eat, even though there's no thermocline to concentrate the food.
Not yet. Not yet. Because there are lots of eels.
Like there are a lot of
I was going to say, there's lots of shallow water eels, the morays you mentioned, but, in those deeper depths down, below 600 metres or so, we've been seeing lots and lots of eels, various great cutthroat eels and basketwork eels, and it's just a lot of eels down there.
Right. Yeah, they're able to swim without using that much energy, that body form factor where they're long and slender. Although we got something else here. This looks like a Siphonophore, I would say. Can you count the number of bells on that? Is it a single bell or two?
J-Rod's saying you can see two.
Okay, so it's a little calicophorine. If it's got rocket-shaped bells, which from the look of the tail, the cyphosome on that, I'd probably guess it's got two pointy rocket-shaped bells, which is the general term for it is a rocket ship siphonophore, maybe a Lensia or something like that. They have two opposed nectophores or swimming bells, and they eject water from each of the bells, and the angle of the bells changes the angle that the animal is swimming at. And all the other zooids that are attached to what looks like its tail are parts of the same polyp colony. And some of the zooids are like polyps that catch food and eat. Others are polyps that reproduce. You have polyps that protect other polyps. Just a range of these polyps living together in repeated units called comedia, and all playing their part to keep the entire colony healthy. Marine snow string. As the snow falls through the water, it tends to take up a comet kind of appearance, with the heaviest part of the particle being at the bottom, and then leaving a streak of particles that like trail out behind it. And then what we can't see is there's also a trail of smell that is left behind in the water as the particle sinks out. And plankton are swimming around until they find this smell of a good food particle, and then they follow it down to the marine snow at the origin, so they can chomp on that. Looks like we still have that calicophorine just went through the screen there. These are probably very small animals. Okay, I think we're cool. We'll keep on going down. Thank you.
So back to the...
I was talking about pigments.
Yeah, great. I'll leave it to you.
So, yes, digesting pigments, breaking them down, making your body transparent uses energy. And as you go deeper and deeper, most of the wavelengths of light have been absorbed, so there's only blue light that penetrates down that deep, which means if you are any color other than blue, and white is bad as well because it contains blue inside it. So if you're red or you're black or you're orange or you're purple, it looks completely black because there's no light of that wavelength to reflect back into the eyes of the animals. So it's actually camouflaged to be very brightly colored down in the deep sea. Looks like we have another Siphonophore here. Is that a Siphonophore? Yes. Yeah, it looks like another Lensia, a little rocket ship. In order to identify these, you need to look at the structure called a somatocyst within the upper bell to see what shape that is. That's the organ in which the Siphonophore stores its food, the nutrients, which it sends down through this central tube, a pipe that connects all the polyps together. So when a stomach catches food, it digests it, and then it shares all of the nutrients that was contained in that food with all of the other polyps in the colony. So it gives some of the nutrients to the swimming bells, which can't eat by themselves. But if they don't swim, then the stomach zooid gets eaten. So they share their food together so they can all play their part. Kind of like communist jellyfish in the original sense of the word. So yes, orange, purple, pink, red, all of these colors are actually good camouflage the deeper and deeper you go. And like chlorophyll, when it's broken down, becomes like a reddish or a brownish pigment. So it's quite common for this reddish brownish pigment, carotenoid pigments, to be left in the tissue of the animals as we go deeper.
Well, we saw lots of these really deep red Medusa jellyfish down near the seafloor yesterday. Many of them drifting past, that lovely scarlet, crimson, purple colour. So really obvious against the white background with the lights.
Right, great. Another calicophorine, it looks like. I find quite a few of them here. So the cameras on the SuBastian are 4K video cameras. So the stream we're seeing on YouTube, I'm not sure if that's 4K or high def. But in either case, it's been compressed to go through the satellite and through the net. When we get these high quality videos back in the lab, we'll be able to play them back in a high quality codec and make out a lot of features that are not visible as we're seeing them now. And hopefully by analyzing this, we'll be able to tell the genus, or if we're lucky, the species of these animals and where they're living. Wow, that's a big discarded Lavation house. Huge packet.
Yeah, that's right, Dhugal. Of course, it's all been compressed. We're trying to get this vision from the seafloor up to the ship, and then to the satellites, down to you, and then you back to us, and then of course it all mixed together with the audio and video. Yeah, it's quite a technological marvel, but it is quite amazing to see the fresh 4K vision on a high definition screen, and there's a lot of detail in there. It'll definitely help you.
Yeah, especially in that darkened control room where you're actually there, and like a second can make the difference between stopping on an animal and not. And that high quality video with the high quality monitors there in the darkened room will really help you to get that job done.
Yeah, the control room is, it's quite large, and it's just wall to wall large screens. It's quite a luxury to sit in front of it. It's fantastic.
Yeah, look at all those monitors.
I counted them the other day. There are 32 monitors in this room.
Wow.
And that's not even counting all the laptops and other computers we the science crew bring in. But yeah, it's like the inside of a TV studio.
You can watch every basketball game at the same time for the season, right? Many marine snow particles. Oh, that looks like, what do we have here? Whoa. What is that? Oh, it's a mollusk. It's like a sea butterfly. Maybe gleba or corolla. Big gelatinous snail.
Like a pteropod?
Yeah, kind of pteropod. But instead of having a calcareous shell, it's got a gelatinous test. I think they're like called slipper. Don't quote me on this. Slipper sea butterflies. Because when you pull the animal out, you're left with this gelatinous test that looks a bit like a slipper or a Dutch clog or something like that, covered in spines. And there's a big, it looks like a Bathycordius house, and another Siphonophore. That very large, massive marine snow we just passed by is the outer filter feeder, feeding filter of a Lavation Siphonophore called Bathycordius. which can make houses that can be 1 to 2 meters in diameter. Oh, there's a Medusa. And the animal inside it's only like 1 centimeter or 2 in length. But by beating its tail, it causes a water current to be produced, which sucks in all the small particles through the feeding filter. Over time, they clog, and the animal has to throw the filter away and make a new one, so it can continue to feed. It's a double structure with a very coarse mesh on the outside that will get rid of all the particles that are too large to fit in the animal's mouth anyway, so that it can't actually eat them. And it will let in all the smaller particles that the animal can ingest. And the inner filter feeder, the inner feeding filter will then trap all the small particles so the larvation can eat them. The last thing we saw was the outer feeding filter, which when they get clogged, the animal will drop it. They make the larger animals 1 per day. Oh, there's a single-celled rhizarian. And what's this? It looks like is that a Siphonophore with a very bulbous cytosome, perhaps? Looks like, yes, this is Agalma okini, probably. As we get closer, we might see some orange. To get closer to the camera, orange color in the cytosome, perhaps. The green fluorescent protein inside it seems to be fluorescing a bit in the lights. Oh, that's nice. Ah. I'm not sure if that is a Agalma okini. The cytosome is extremely bulbous. What kind of a size do we think that might be? Did the pilots have a guess based on its movement?
They're saying about 10 centimetres.
10 centimetres. Okay, could be a Agalma.
Dhugal, just going back to these larvaceans, the fact that they're creating these big external houses which they're discarding all the time, how that must take a lot of energy to continually make all that jelly. Is that, got any comment about that?
Yeah, and the really cool thing is the houses are actually made of cellulose. So these animals make animal-based cellulose. Cellulose is the material in plant cell walls that keeps everything hard and gives us wood. But the tunicates make it to make their bodies hard as well. And the larvaceans, they're called larvaceans because they look like the larvae of regular tunicates or ascidian sea squirts. And they use this to make these houses. The fibers themselves are very thin. And although the size of the house itself is very large, there's a lot of space in between each of those fibers. So the actual organic matter input that they're putting in compared to the amount that they're able to take out of the water through the filtration mechanism, you do the sums and it's a plus for the animal.
So they're just pulling that they're just pulling the cellulose out of the water column, I guess, from the phytoplankton around them. Is that right? They're just harvesting that?
No, they're making it themselves, we think. It's a different chiral form. You know, with molecules, you have like a right-handed form and a left-handed form. And most of the enzymes in the world have evolved so that they will fit one of those forms, the form that is most common. I forget if it's the right or the left right now. But the cellulose made by the tunicates is actually the other type, so it's not easily digested. And we still need to do work on finding out what kind of animals can digest that cellulose, because there's a large mass of carbon being transported down into the deep sea to the sediments, which should be available as food if anything's got the enzymes that can digest that particular chiral form. they've actually found even in human skin there are small amounts of animal cellulose. So it seems we're able to make it too. Or it might be one of our symbiotic biota that makes up part of the human body. We still don't know. Here's another Siphonophore. Ooh, ooh. Once we get a video of this, can we suck this guy up, please? I wonder. We're still very shallow for this to be Rodgicovia, which is has been found in the Antarctic. Cyphostome is a bit different. It could be a halostema. I don't know what this is. And the nectosome has got these 4 white spots in it, which might be parasitic amphipods or something. There are very few stomachs for a Siphonophore, very widely spaced out. It looks like some of them might have something in them. So if we catch this guy, we'll find out what he is, we'll find out what he's eating, we'll find out what his parasites are. And because we know where he lives at 242 meters depth, we'll know habitat preferences such as temperature, salinity, oxygen, etc. There's actually a record of a Siphonophore similar to this that was feeding on eel larvae. And although the stomach was only like a centimeter or so in length, it had eaten an eel larva, which was something like 12 centimeters in length, because the stomach just expands, and it had folded the eel larva over so it would fit in. Really amazing stuff. Yeah, very nice. A Physonecht Siphonophore. This is different to the little rocket Siphonophores we had before because it's got a float called a pneumatophore, which is full of carbon monoxide, which is normally, it will kill you. But the structure of the float of these animals is, rather than having endoderm, which is the inside of skin, it's turned inside out, so it's got ectoderm, the external surface of the skin on the inside of the float, and that protects the tissue inside from being poisoned by the carbon monoxide. And it uses that carbon monoxide as a gas, pumping more gas in to help it float shallower in the water column, letting gas go as little bubbles to make it go deeper in the water column. So these guys often vertically migrate, heading up to the surface at night to eat, and going deep again during the day. And these little floats full of gas are very reflective in the sonar. So as ships were going along looking for submarines and things, they found these large scattering layers in the midwater. And one of the major animals that was causing these scattering layers were these jellies, the Siphonophores, which when you drag a net through the water, you don't catch because they break up into little pieces or are strained through the mesh. But here with an ROV like the SuBastian, we can suck one up as if we're eating noodles and capture the animals. Hopefully.
Seafloor, single beam echo sounds would see like the seafloor suddenly rise up, and everyone would hit the brakes thinking about to hit an island. And nautical charts are covered in these things called vigiers, right? Sort of like an unknown feature on the deep ocean. And in a previous life, I was a hydrographic surveyor, and we had to go out and resurvey these vigiers, and in every case, it was just deep water. So they were just recording these deep scattering layers or plankton rising to the surface at dusk each day.
Yeah, so fish swim bladders and Siphonophore floats, I think, are the major culprits. Oh, he's still there. That's good. Must be very small. Lisa?
May have lost it. Yeah, unfortunately. There's a bit of a current here.
Okay.
Yeah, we're blown right off course, Dougal, so we'll just need to come back.
Roger that. Come back and get in a position for observing while I put on my blindfold and try not to yell out, "Wow, I haven't seen that before." So you can get more maneuverable for the next one. Yes, the ROV is attached to the ship by a tether, which is sending down power, and it's also sending the comms, the communications, down to the robot, back up to the surface. And the tether is only a certain length. And if we stay on an animal too long, especially in the shallow areas where there's not that much cable play in the cable, it's very, very hard to maneuver the ROV for any amount of time. You only get like a very short amount of time on every animal where you can move the ROV without trying to pull a ship along behind you. It's very, very difficult to do surveys of the midwater at these shallow depths. Arrowworm or Ketignath. I'll just keep one eye open. So we're driving the ROV so that the marine snow particles are coming into the camera. This way, even if we're going a little fast, we know that every particle is going to be in focus for at least one frame as we drive through it. Kind of like collecting insects on the windscreen. And at that sweet spot, which is, in our case, the field of focus, where the camera is in focus, when you're driving along, the sweet spot's the windscreen itself, which will, the bug stops on there, you get a good look at what it was, whereas you couldn't see it when it was coming towards you before it hit the windscreen. Well, we just jog through the video frame by frame until we find that sweet spot where the particle is in focus. And so we're going to be looking through all of these particles later to try and tell which ones are living particles, which ones are dead particles, and identify the living ones if we can. It can take 4 hours, sometimes more, to analyze 1 hour of videotape. So we've got a lot of good material here that we're collecting in terms of video footage. And it's not just one camera either. We have multiple cameras set up in the water, all looking at different things, different resolutions, different fields of view. Some are very wide fields of view so that we can image entire animals or image the environment around an animal, which we're imaging with a zoomable, focusable camera, which we're using to get those really tight shots of the animals themselves.
Dougal, do you want to mention your own GoPro cameras that we've also got set up on the ROV?
Yes, so this camera that we're streaming now is the 4K main video camera, and it's set up on a pan tilt unit, so it moves left and right, up and down, and the field of view will change depending on if you're zoomed in or coming out. And this is really great for following an animal through the water and trying to get good footage to identify what the animal is. But sometimes you also want to know, all right, where this animal was, what was the concentration of food particles? Is it there because of the temperature? Is it there because of the salinity or the oxygen? Or is it there because that's where all the food is? And so in order to capture this, what we're trying here is we have some GoPro cameras, which have a very wide field of view and a fixed focus. And we have them in a stereo configuration, 2 of them in the same housing, in order to map which pixel in the left camera equates to which pixel in the right camera. And if we know that for all of the different pixels and we calibrate the cameras, so we have an object of known size shown in both cameras at the same time, we can then use those images to measure the sizes of the animals and also the distances between different particles. And by doing this, we can get quantitative data on the number of particles, food particles in the water, or the number of animals and their sizes. We also have these GoPro cameras, single cameras, giving a wide field of view facing downwards to look at the seafloor, and forward facing as well. So we have, we're looking at, we're recording multiple angles in a wide field of view to get an idea of the environment that all the animals are in that we're videotaping with this 4K camera.
Dougal, we're experimenting with doing photogrammetry with a high-res, the 4K vision here. Like we can take still images over the seafloor and create full 3D point clouds of the seafloor. Can you do the same with this midwater cameras? Can you create a 3D model of a large plankton with your cameras?
So the software, this is like structure from motion photo scan kind of software or proprietary stuff perhaps you've been developing. Normally it assumes that there is a seafloor and that you cannot see forever in front of you until the light peters out. So it's not quite as easy as it is with the benthic stuff. We're trying to develop a way to do it now. One technique we've also been using is in combination with the stereo cameras. We have a line laser. So we're getting optical slices of the water, and every time it's a green laser. So every time a particle flashes green, it means it's at that particular distance from the cameras. And we're able to use just the green parts of the image and put them on top of each other to try and scan through the water in 3D. So that's what we're working on now. The only thing that's proving difficult is the ROV is not always going at exactly the same speed in the same direction. And the animals tend to swim or move as they're being scanned as well. So we have to stabilize the images first before we can get the 3D reconstructions. And stabilizing the images is hard when there's no stable point of reference as there is with like the seafloor, because you can't just pick another particle at random and assume it will be stable. So it's technically very challenging.
Yeah, I'd agree with that. Yeah, J-Rod was just saying they've done some work with Mbari, the Monterey Bay Aquarium Research Institute, where they've been using lasers to kind of scan. But you're right, you've got to get your animal still or scan it extremely quickly. So when we do photogrammetry on the seafloor, if you've got fish moving around across that seafloor, you cannot really create a decent picture of a fish. You can of the seafloor, of course, because it's not moving, but any marine life moving over it just becomes this blur and is excluded.
Yes, another Siphonophore, Calicophorine, it seems. Yeah, I've been doing laser scans with high-speed cameras, taking hundreds of frames per second to reconstruct jellies. But you can't send that kind of bandwidth up the umbilical of an ROV because it's just so much data. And you can only get like a second or two at a time, usually. That's Kakani's group at Mbari who's doing those scans. They're doing it with a red laser, very high power. And they're getting 3D reconstructions as well of the animals that are, usually the animals that stay nice and still.
Yeah, J-Rod's saying that's what they've been using. They've been helping with those guys, and that's the system they're using.
Okay. Yeah, that's really exciting stuff. If that can be developed, and we can actually see the animals from all angles in that case. Here's another eel larva. Cool. Yeah, there's so much we don't know about the midwater. It's the largest biome on the planet for animals that aren't bacteria or archaea, like any multi-celled animals. land is just a very thin membrane on top of the rocks and dirt. There's things that live in the dirt, but not all that deep if they're multicellular. The surface of the sea, which is where we normally have access to, is a very thin layer where the sunlight's penetrating. The seafloor also, okay, there's topography, but it's also very small in terms of volume when you compare it to the midwater itself. And the number of midwater surveys that have been carried out, very few. The number of researchers who are involved in this kind of work is very low as well. And so this is like one of the last frontiers on our planet for making new discoveries about what's here sharing the planet with us, which is really exciting because every dive you find something new that's never been seen before. Keep you on the edge of your seat. Oh, there's a medusa. It's like a hydra medusa. Where is he? Looks like it's a halocreatid. This might be the haloscera that we tried to sample previously. Once we get some video of this, I'd like to catch this. Please. You'll notice the tentacle tips are whitish. They're like solid rods covered in stinging cells, whereas the distal areas, or the areas that are closest to the bell, are more transparent. That's a character you can use to tell the halocreatid family of medusae from other hydromedusae. We still don't know why the tentacles are like that, and it's a very difficult character to use for net caught specimens because they normally lose all of their tentacles. They throw them away like a lizard throws away its tail when you try and catch them in a net. These animals have a very rounded stomach, just like the Narcomedusae, which is another very unstudied group. Oh, nice. You see how many radial canals are on there? Are there 8 or are there more? If there's more, it'll be the genus Halotrephes. If there's 8, I'd say it's a Haloscera. We should be able to count them with the high quality video later. Doesn't look like the Halotrephes we caught off the Cairns Seamount. I think it's something else. And it'll be a very nice sequence to put in the tree to try and tell exactly how these guys are related to each other and how they evolved. Ooh, he's really small and close. So they have these special tentacles that are different from all other jellyfish, but we don't know what they eat. You never find them with food in their stomach. You catch them in a net and the stomach is torn open and whatever they've eaten has come out. And even with an ROV, I don't think I've ever seen one with something in its stomach. So these animals, you find them all over the globe, and we really don't know what they eat or what eats them. With this animal, when we catch it, we'll take a couple of tentacles and preserve them in ethanol to do DNA sequencing. And we are hoping to use this to make a DNA barcode database so that we can probe the stomach contents of various animals to see what these guys are being eaten by. So that's one of the things we're doing during this survey, is to get good quality video of animals to train an artificial intelligence machine learning system and get the DNA barcodes for them as well with a correct taxonomic ID, so that other scientists who come along afterwards who maybe have caught a damaged animal, which they can't tell just by looking at the specimen itself what it is, will be able to sequence what tissue they can get to be able to get a proper ID with it. Keep our eyes out for another one if we find one. We're getting down close to 300 meters now. You'd normally expect krill, little shrimp-like whale food, also jelly food, to be turning up soon. These are the kinds of depths that diel vertical migrants will be inhabiting during the day. Probably still quite warm though, there off the coast of Australia, I would imagine.
Yeah, it's 15.3 degrees C now, at hitting 300 meters down. So we're still what we believe is subtropical mode water. Around that 400 meter mark, we'll see a definite thermocline to start getting colder much more quickly.
All right, so the migrants may all be down below 400 then, keeping nice and cool and refrigerated, conserving energy.
Yeah, definitely. Yeah, that 400, 500 meter mark is quite a distinct thermocline around here. All that colder water is sort of trapped below that 400 to 500 meter mark.
Okay. There'll still be animals living at these depths because a lot of animals hang around above the layers formed by the diel vertical migrants during the day, so they can hang out there, and as the migrants pass through going to the surface at night to eat, they eat them. As they come back again, they eat them again. So they just hang out, not moving very much, trying to conserve energy in the slightly warmer water, just so they can hit those guys and eat them as they go up and down.
Dougal, have you seen krill down these depths on previous ROV dives in this part of the Ribbon Reefs?
Yes, we've seen krill. I'm not sure if they're quite this shallow, but between 300 and 400, I think they first started kicking in on other dives. Euphausia krill, normally you can tell they donut. Their swimming is kind of donutting like this, or they spin on their axis, looking up, and they're just spinning around and around. They're the two normal behaviors for euphausia. There's another group of krill that have bilobed eyes, which they kind of swivel so that one facet, which is evolved for looking up at downward welling light, can see what's passing above them, looking for shadows or whatnot. And the other lobe of the eye is looking out for predators that are in turn looking at the krill, trying to find them against the backdrop of the downward welling light. And so they can pick out the bioluminescent point sources produced by the predators. And they normally actually swim upside down. So if you just catch the animals in a net, you think they're swimming with the legs pointing down, but I've never seen them swimming like that. They're all swimming upside down with the back pointing down. It's kind of cool, something you could only see through in situ observations, which changes your interpretation of the eye structure when you know they're doing everything backwards to what you thought. Oh, there's a krill. That was a krill just then. What else do we have?
Yeah, J-Rod's just doing a bit of repositioning of the ROV, the ships to the west of us pointing towards land, and there is a decent current, so just a bit of manoeuvring here, Dougal.
Roger that. I'll try not to suddenly yell out giant squid or anything.
We'll stop for giant squid.
Okay.
We have a list now. It's an ever-growing list of things we'll stop for. I quite like looking at the giant isopods, but yesterday we hit an octopus and also Dumbo octopus, so we stopped for them. But when we're out in the Coral Sea, we'll stop for nautilus as well.
I noticed you had a nautilus with a very strange pattern on the shell.
Yeah, we saw quite a small nautilus. One of our, I think it was about 2 days ago when we were closer to the reef in about 400 metres of water. So, but out in the Coral Sea, which is tomorrow, we're heading out further in the offshore Coral Sea area tomorrow. We expect to see quite a few more nautilus.
Was that a very small nautilus over a sandy bottom, perhaps?
Yeah, it was. It looked like it was sleeping. We've come across them before. They've closed their operculum up. They're just resting on the seafloor. So yeah, to my sense, I think they look like they're sleeping.
All right. I remember the only baby nautilus we saw during the Picasso expedition to Osprey were also on a sandy bottom, which was completely opposite to what I thought they'd be doing. I thought they'd be hanging out and hiding behind rocks or somewhere.
Yeah, that's right. We were quite curious what was the habitat, but I think ultimately they're scavengers and they're attracted by the light or sound of the ROV. They're always fairly mobile, except for the occasional one that we find just resting down on the seafloor.
I was hypothesizing that they might be like eating little polychaetes and things in the sediment, because if they went to food falls when they're still so small, they might be eaten by something else that went to the food fall. Because you don't catch them in traps that much, do you? The juveniles, or do you?
I don't know. We might pose that to Dean Miller, who's our media person here on board. He spent years working with Andy Dunstan to study these nautilus, so he's got some tales to tell. When we get out to Osprey Reef tomorrow, we might get Dean on and talk about his experience trapping nautilus for scientific studies.
Roger that. Look forward to that. How's their position coming along?
Yeah, it looks like we're about to settle back in, so we might be able to slow down a bit for you. So getting close.
All right. This is dive 388, is it?
Yep, this is ROV dive 388, and each ROV dive has its own unique number. Even if it's aborted for whatever reason, like there's a technical issue, it's still given that unique number, and the next time the ROV goes back in the water, it's given the next number. Just a quick answer to question, how large is the nozzle of the sampler? That suction one that we've been using for the jellies is about 75 millimetres across, so 7.5 centimetres.
Cool. Yes, some fairly large objects around. Oh, looks like we got one there towards the bottom left of the screen. We're stopping on currently. Oh, is that a saw missus? Looks like it might be a dinner plate jelly. $378 or 9 meters depth. There we go, the tentacle count there. Looks like it's got quite a few tentacles. So this is a genus of Narcomedusa, which most of their food is other gelatinous animals. And we're currently working on the taxonomy because a lot of different species have all been lumped together as a single species up until present. And we believe that there are multiple species. So we're recording the habitats of the animals, their morphology, so we can tell them apart from the specimens themselves. And we're also collecting DNA samples, tissue samples for DNA work to tease apart who's related to who and who's actually the same species as someone else and who is not.
Dougal, do you want to sample this, or are you happy to move on? What do you want?
Yep.
Yep.
It will be another piece in the jigsaw puzzle.
Slipped up.
Awesome.
Have we got James online? Welcome aboard, James.
Jamie Roma? Okay, we've just caught a soul missus. Oh, there's another Siphonophore, a Galma okini, probably. Physonex Siphonophore. Seems there's quite a few animals around. Any more than I'd originally been expecting. Every dive when we've been doing these shallow depths, we've been seeing quite a lot of life. I was expecting it to be more of a desert out here, so this is pretty exciting. I guess most of our dives have been around canyons or seamounts or places that are high points of productivity, and perhaps that is why.
We're right, we are at the downstream end of the South Equatorial Current, so this is oceanic water for all intents and purposes. So my thinking is this has been swept out of the Southwest Pacific.
Yeah, we did some work in the South Pacific Gyre a year and a half ago, and the water was almost completely blue. You couldn't see any marine snow particles most of the time. But that was right in the middle of the most oligotrophic place on Earth. Certainly a lot more happening here.
Yeah, this is the very start of the East Australian current. So that South Equatorial current comes in, it sweeps over the Queensland plateau, and it basically arrives just about this latitude where we are now. There's a northern arm, comes the Hiri current or Coral Sea countercurrent, and that sweeps northwards up towards Papua New Guinea. The southward moving arm becomes the Great East Australian current, the EAC, which was in the movie Finding Nemo, and that sweeps all the way down the outside edge of the Great Barrier Reef as far down as Sydney, sometimes spinning off little gyres that can be, you know, almost as far as Tasmania. So it's a really huge three-dimensional river. We are right in the sweet spot, the very head of that EAC current, where we are at the moment.
Great. So some of the animals we're observing here will probably be entrapped in the gyres moving south and providing food for the animals that are living down there as well.
Yeah, absolutely. They're right at the very start of this great big river that's about to take them thousands of kilometres further southward.
And not many of them will be caught and put in the dentist's aquarium. Wet fluid house, larvation house there. Oh, coming fast and furious. Couple more medusae, I think I saw.
Yeah, we just got this inset up. You can see this is the 3D model that we've been mapping through the night. So you can see we're right at the base, well, about midway along this very, very deep canyon at the northern end of the Ribbon Reefs. So if you can imagine the EAC I was just describing, it comes in from behind us from the east. It sweeps in towards the Great Barrier Reef, splits, the southern arm becomes the EAC, and it just continues moving along this great long margin, just the Great Barrier Reef.
Oh, wow. What do we got here? It looks Siphonophore-like. Could this be a thalassocalus? The sea cup or the mask comb jelly? If so, it would look like a rice bowl, a bowl, which is a very strange shape for a Siphonophore. It looks at first glance to look like a regular medusa. Certainly looking that way. Yes, it is. Yes, that's the Thalassocalus inconstans. The gonads don't seem to be as developed as the ones we'd seen on a previous dive. Those lines that are looking like the eyes in a mask. Imagine that the animal itself is a mask, and these eyes are actually the gonadal tissue. Siphonophores are hermaphroditic, which means they have both male and female sexes in the one organism, and they can even reproduce themselves. So if you get one of these animals in the ballast water of a ship and it's released into a new ocean, they can start up a completely new population of comb jellyfish just from one single animal, which is what happened in the Black Sea to Siphonophores that have invaded the Mediterranean now. Very nice. Can't see any krill in his stomach, but they do eat krill. You can see the lights that are being reflected from the ROV lights as the combs change angle as they're paddling through the water. They're reflecting the lights of the ROV. And the color changes depending on the angle of the combs and whether the combs are sitting on top of each other or they're single. And the light's interfering. If you look very closely, you should be able to see some canals running through the jelly of the animal. These are supplying nutrients to the larger gelatinous portions of the body. It's got 2 small tentacles hanging on either side of the mouth. You can see in the middle of the animal, there's a tongue-shaped structure. This is the pharynx, or stomach of the animal. The mouth is at the end, which is flanked by those 2 white spots, which is the tentacle bulbs, from which 2 tentacles hang down into the bell. Thank you, that was very nice. Oh, and there's another one. That's a lobate, though, different kind of cone jelly. Alrighty. Let's set ourselves up for another observation. There's a krill. So you really have to concentrate to see these very transparent animals approaching from the distance in order to stop on them in time. Takes quite a lot of concentration and experience, of course, because of the lag we have here with the images coming to my video screen. The pilots have a lot of experience flying here in the midwater and finding targets that they think are interesting. So they're stopping on the animals, and by the time I see them, the ROV's invariably already stopped on the animal and is taking a look, which really helps because otherwise those 2 seconds really devastating works.
Yeah, Dougal, just looking at our time for today, I reckon another 2 hours on this dive should keep us about right on target for then going deeper with the benthic component. How does that sound from your perspective? Another hour to get
Roger
to
that. Let's keep going down there.
yeah. Another 2 to 2.5.
Okay. I'm hoping we can make 1,000 meters at least.
Yeah, indeed. Yeah. Well, actually, 1,200 would be great, but yeah.
Yes. Oh wow! Thought I saw a couple of Siphonophores go past there. Might just be my fingerprints. Possibly a Siphonophore there. Looks like there were more animals shallower than there are just here. It's interesting. This is where we were expecting to have larger amounts of animals because of the colder temperatures. Maybe there aren't that many vertical migrants. It might be too warm in the surface waters for them to be able to survive up there. And they might just hang out down here to eat stuff that falls out from the surface later.
Yeah, we're probably getting close to that big thermocline between the warmer surface waters and the cooler stuff. So we're now down to 11 0.2 degrees C, so it's definitely cooling now. 443 meters deep.
Roger that. Still haven't seen any animals with red pigment, suggesting we're still getting downwelling light, at least amounts that the animals that are used to living here can see, although we would never pick it up on our cameras or by the naked human eye. There's a krill. Although krill look to be, they look like shrimps. You can tell a krill from a shrimp because it has gills coming outside the body in between its legs. Whereas a shrimp, their gills are actually inside the carapace in behind the head. A good way to tell krill and shrimp apart. There's a fish, possibly a cyclothone. A salp, solitary salp zoid. Oikoplurid, lavation. Ooh, a little solmissus perhaps. You videotape him and slurp him up if we're able to. You're too close.
Yeah, it looks like the guys are manoeuvring now to see if they can get it.
Yep, it's got stomach pouches. It's definitely a solmissus. This is what currently is known as Solmissus incisor, which is a species complex of multiple species, which we're trying to tease apart.
Is that some food inside? I see a little white dot
Could be,
inside.
Yeah, it looks like, you may have eaten something. Or it could be an amphipod or something parasitizing it. See if he's embedded in the jelly or actually in the stomach.
Here comes the slurp gun.
You went in?
Yeah, we got it.
Excelente. Well done. And there's some krill. On the left of the screen, a krill, and we're panning the camera around to see where that solmissus is and record the jar number. There he is, canister 2. Yep. SOLMISSUS. Solmissus. Okay, very cool.
Seeing a lot more krill down here now.
Yeah, so it does look like they are migrating down into the colder waters during the day. But they do seem very active as well. It could just be because of the ROV lights causing them to react and come out of their sleep, their daytime sleep. Kind of hard to tell if that's the case when we have to have the lights on for us to be able to see. they actually found that the numbers of krill that you catch in a plankton net increase if you flash bright lights at them, because they tend to act like deer in the headlamps and just freeze for a minute and or a few seconds. And while they're frozen, the net can collect them. Otherwise, they swim out of the way of the plankton net, and you don't get truly quantitative data. I remember doing dives with Picasso where so many krill were hanging out in front of the camera that we couldn't see anything but krill, and we had to turn off the white lights and switch to the red LEDs, and the krill couldn't see the red wavelength, so they all lost interest and left. And we cruised around with these red LEDs for a little while and came across some fairly large fish. We turned the white lights back on again so that we could see what kind of fish they were, the color patterns, etc. And as soon as the white lights came on, all the fish went, oh, and darted away because they could sense the white lights, but not the red.
So, Dougal, you've been to Antarctica a few times, and so have I. You and I were both involved with the census of marine life, and you went south on a Japanese ship and went south on an Australian ship. But, of course, you would have seen lots of krill in Antarctica, which is the basis of the food chain there, isn't it?
Yes. Yeah, many, many krill. It was a bit, for the jellyfish, it wasn't that nice because there were so many krill. And if the jelly gets caught between a mass of krill, there's not much left of it when they come up. And there was certainly a lot of krill down there. Usually the krill and the salps eat similar kinds of foods. So at least in Antarctica, they don't tend to occur in the same place so much. I noticed we saw salps more shallow here, whereas we're seeing the krill down at this depth. Wondering if there are salps here in the same layer as these krill or not. Haven't seen any yet. What's that?
Yeah, Siphonophore.
Beautiful, okay.
Do you want to stop for Siphonophores there, Dougal?
I do, yes. Just to get video. The samples won't survive preservation, but with 4K video, if we get a variety of angles, we can tell the species. And there will be so many new records for the Southern Hemisphere just because nobody's looked at deep water Siphonophores with an ROV down here before.
Yeah, copy that.
Larvation house. I haven't noticed that many rhizarian or single-celled planktonic organisms, which can be quite common in areas without so many nutrients. There seem to be like lots of jellies and tunicates. So more food around than there is in the South Pacific Gyre, I would say. They're probably out there, and I might just be concentrating on the jellies because they're bigger and know more about them. So when we go through this video later, we'll be having a very close look at all of the particles so that we get true quantitative data rather than kind of guessing at things. Almost at 500 meters depth. So we'll be expecting probably Colobanema to turn up soon. A Trachymedusa, another hemispherical jelly that drops its tentacles, glowing. Oh, there you go, a coelodendron. So that's a single-celled, speak of the devil, and there he was, a single-celled rhizarian zooplankton. Some of these guys are like, they look like little transparent sea urchins made of glass. And you'd expect a single-celled animal wouldn't really eat anything very big. But these guys, they put their pseudopodia out of their test. These are larvation houses here, these two. And the pseudopodia grab onto things like copepods. And like they wrap the pseudopodia around these copepod legs, and then they pull the copepod onto their spine. So they wrap their pseudopodia on, and they pull the copepod on and impale it on a glass spine. And then other pseudopodia come in and digest the copepod. And when they finish taking all the nutrients out of it, they push him off the spine he's been impaled upon and let the carcass sink down into the ocean's depths. So they may be single-celled animals, but they are voracious predators, and they can eat other animals larger than themselves. Oh, here's a ctenophore. This looks like it might be Thalassocalus again, the sea cup ctenophore. Yes, I would say it is. Quite deep for, have a look at the gonads, see if it's mature. Oh, there's something else at the top of the screen there. What's that? Oh yeah, let's look at this. It's a lobate. Let's take a look at this guy. Is it flattened, and does it have triangular projection? Whoa, it's swimming. It's an Oscillopsis or a Bathysauroi. Follow that guy. Which? It looked like an Oscillopsis.
Yeah, they're trying. There it is.
There he is. So we'll be able to tell if we can see tentacle bulbs at the top of the stomach or not. It certainly looks Oscillopsis-like in the fatness of the main part of the body. Yeah, cool. I think I should be able to tell what it is just with that video. And any more you can get is good. There's krill there too. You make out the two white spots that are the tentacle bulbs? Doesn't look like the gut is pigmented. I want to say it's Oscillopsis, but the way it's swimming is very Bathysauroi-like. Yeah, I think I can get an ID from that, so thank you. There's quite a lot of animals that, through convergent evolution, look quite similar to each other down here, and you really need to see the details of the body structure, because at first glance they look really similar to each other. There's mimicry as well. You can get things like eel larvae will wrap themselves into a ball so they look like a jellyfish, and a predator doesn't want to eat the jellyfish, although they love to eat the eel larvae. And so in the dim light, they see what looks like a jellyfish and pass it by, when actually it's a yummy little eel larva.
From J-Rod, have you ever seen a squid that looks like a Siphonophore?
Yes, there's a family of squid, the Chirotutidae, where the juveniles have a very long pen which is attached to their backside, which in like cartoon versions of squid is where you'd call it the head, I guess, where the arms look like legs. Anyway, hanging out the back end of the animal opposite the eyeballs, there can be a really, really long structure. And this is a mimic for siphonophores. Even the way the carotuded squid hold their bodies as they're floating around, because the siphonophores are angled at different angles depending on what depth they are, if they're actively feeding or not. And the carotudeds angle their body in the same angle. So it's seems to be a really good case of mimicry in the midwater, as I'm sure J-Rod knows since he asked that question. Someone?
Yes, it was a planted question.
Chain of salps here. 1, 2, 3, 4, 5 zooids, it looks like.
Hey Dougal, Blair on Facebook is asking, do you have any other imaging systems mounted to look at the plankton in more detail? Maybe you want to talk about your GoPro setup rig that we've got on SuBastian.
So right now on the SuBastian, we have an array of different cameras. The 4K camera, which is imaging now, which we use to zoom in and look in fine detail. Oh, is that a phronomer in a barrel? This might be a pram bug, a hybrid amphipod living inside the hollowed out carcass of a pyrosome tunicate, which it like scrapes away at the surface, and the tunicate is being eaten alive, so it keeps excreting all this cellulose to try and harden the body up so it doesn't get eaten, and that hardens the body and makes it so there's no cells inside. So you get a mostly acellular matrix, which, because there's no cells there for bacteria to digest, can survive for very long periods of time in the cold midwater depths. And inside here, we see a female amphipod, a little crustacean like a shrimp, which is living inside there. These animals plant their babies usually on the wall of the barrel, and they swim around. Right now, it must have realized the ROV's here, so it's pulled its entire body into the barrel and is kind of hanging out, hoping we'll go away. But normally, they hang with their claws and their tail sticking out the end of the barrel, and they spin around, and they knock out jellyfish. And as the jellyfish turns over, it gets the tentacles out of the way, and the phronomer rips at the ex-umbrella of the jellyfish and rolls the tissue up into little balls and sends it down into the barrel for its babies to catch and feed on. The face of the head of these guys looks like the alien queen from the movie Aliens with Sigourney Weaver. It was actually, the alien was modeled on this amphipod. So it does look very similar. The top surface of the head is a light gathering eye, and the light comes down in a prism to a very, very small area that has pigment, because these guys want to stay as transparent as possible so that they're not seen by their predators. So the area with pigment is very small, but the area gathering the light is very large. And this eye surface would be damaged greatly if a jellyfish tentacle hit it. So they normally keep their heads inside the barrel, even though they hang the claws on the outside so they can catch the jellies for food. So he's sitting inside this barrel with his arms, with her arms, sorry, pushed out against the barrel so it doesn't get dislodged and pulled out of the barrel. You can see all these little bumps on the external surface of the barrel. This shows that it was a pyrosome, a fire tunicate, a bioluminescent tunicate that it fashioned its barrel from. Different species of this family of amphipods use different animals. They can use salps, they can use siphonophores, etc. to make their house, but the bumps on here show that it was a pyrosome. Very cool little animal. Doesn't look like it's got any babies there, Dan.
Yeah, very cool. We're really impressed here, and also the thanks to the pilots for keeping everything in focus. It's pretty remarkable to see this detail.
It's nice. Chronoma sedentaria, probably. Don't think I've ever seen an in situ shot looking down on them like that. That's cool. Okay, let's keep heading down and make sure we make 1,000 meters. Yes, so this main camera is for getting images like that. You'll notice as we were zoomed in very tight on the amphipod, we wouldn't be able to tell if a giant squid swam by close by. There are other cameras on the Sirius, which are situation cameras, so the pilots can see what they're trying to sample from different angles to get an idea how far away they are, for example. We've also set up some GoPro cameras in special pressure housings that have a very wide field of view, and the focus is fixed. If you give a camera to a scientist, they tend to play with it a lot to try and get their best shots of their favorite animals. And if you do that, the data is no longer quantitative in terms of how many animals were where. So we've got these GoPros on there capturing the wide environment, so we can see what's going on and how many particles there are around the animals that we're filming, if there are predators or any prey items around there as well. So while we're zoomed in on, say, that Siphonophore before, the GoPro cameras are recording how many krill there are in the vicinity. So we can compare to say, in the places where there are lots of krill, is that where the Siphonophores are? Or do the Siphonophores eat all the krill and therefore there's none there? Or are they more associated with copepods or things like this? Although we don't actually have them on SuBastian, we have a range of other cameras we use to image midwater animals, including shadowgraph cameras, which project an LED light, projects light through the animals, which is picked up by another camera facing into the light. And the light's completely collimated, which means all the light rays are parallel to each other. So it doesn't matter where the animal is within that volume, they always look to be the same size. Normally, if you have a camera and the thing you're looking at is close to the camera, it looks big. And if it's far from the camera, it looks small. So having collimated light in the shadowgraph gets rid of that problem because it looks the same size no matter where it is in relation to the camera. Unfortunately, because of COVID-related travel restrictions, we were unable to get those cameras and the people who can operate them there on the ship. So we're doing the best we can to try and get like a variety of different images to give us more of a holistic view of what's down there. Some of the GoPros are in a stereo housing because on the seafloor, it's common to use a pair of lasers to project on the seafloor, say, 2 laser points 10 centimeters apart, which will help give you a scale for the size of the animals you're seeing. What's this? Is this the self chain, it looks like, perhaps? Yes, self chain. Maybe the genus Selpa, since they're organized like this. And that's in an arrowworm, a Chaetognathes. In the midwater, you can't use these laser lights usually to get sizes of animals because the animals are so transparent, the light goes straight through. You have to be very skillful and or lucky to hit these 2 lights on a reflective surface on the animal you're trying to size. So what we use is with the stereo cameras, we map out which pixel in the left-hand camera is the same as which pixel in the right-hand camera, and then we click on the same pixel in both screens using some special software to try and say, okay, how far apart is this pixel from that pixel? And that way we can get an idea of what the length of a fish is, or the diameter of a jellyfish, for example. And we have those systems on the SuBastian. Oh, what do we got here? Are we able to go in for a closer look, or do we have a cable? length problem.
Oh, we may have lost it. Oh no. Yeah, we were looking at something else. This could be a larvation house, perhaps. Something in the middle screen now.
All right, that white. Yeah, I see him. I'm not sure. Let's take a look. Yeah, it's a larvation house. Oh, or is it? I think it is, yeah. We'll be able to tell from the video.
So, Dougal, all of those GoPro cameras, I mean, to use them effectively, you need to have them calibrated, wouldn't you, against a frame?
Whoa. Is that a lobate? That's a weird one. Hey, come to daddy. What do we got here? Does it have oracles? Oh, that's what we were just trying to chase before. Same species. This one's being a bit more cooperative. Nice. Oh, wow. I'm getting for as many angles as we can. Where are those tentacle bulbs? And the shape of the gut will also be helpful. That's nice. Okay. We should be able to ID that.
So, Dougal, the beautiful colours you see on the cilia, I know they're reflecting light, but can you talk about the different, like, why are we seeing all those different colours shimmering along the swimming cilia?
It's kind of a combination. The cilia are fused together to form a comb plate. So it's a single flat object, which is actually made up of lots and lots of different fibers. And some of the light is like reflected, and it depends on the exact angle at that moment in time where the cilia is. Another thing is the cilia overlap on each other. They're not like this. They're more like this, and you can have, depending on the species, 2 or 3 cilia that overlap each other. And if there are different thicknesses of each of those comb plates, it can react with the light in a different way and give you a different color. So you kind of end up with a rainbow of colors, because as the animal is moving its cilia, you have huge numbers of combinations of different angles and overlaps, which tend to give you lots of different colors. The animals themselves bioluminesce usually with a blue light.
Yeah, we're just putting a light down from above.
Yeah. We know that at least some ctenophores have the enzyme to break down the substrate that makes the light, but they can't produce the substrate themselves. So the copepods and other animals they eat, the ctenophore absorbs those molecules that those animals are using to make their own bioluminescence, and uses them as their own substrate rather than using them for energy, which is pretty cool. Yeah, that's great. We should have enough video from this to be, oh yeah, nice. Well, I think it's an Oceropsis. It's very deep. Awesome. Thank you.
Yeah, temperature's dropping fairly quickly. We're at 8.5 degrees C at 560 metres down.
Okay, right. Yeah, that's a big temperature drop. So being able to find these animals at what I'd assume is the edges of their ranges informs us of exactly what temperatures or what salinities or oxygen levels are they able to survive at. And obviously that guy was in quite good shape. It was able to swim and try and escape. It didn't look like it was energy limited at all. So it's not here because it's sunk here because it's about to die, or it's been transported from somewhere else and is only just hanging on at this temperature. It looks like this is a habitat that it's quite happy at being at. So we'll be able to identify that animal to species and be able to say definitively they can handle temperatures up to this. And we're trying to collect this kind of information for as many species as we can to be able to get, well, not guess, but to get an estimate of what will happen as the oceans are warming, or as the pH of the oceans is decreasing, or the oxygen levels are decreasing, what is going to happen to the distributions of these various animals. And are we going to have prey that no longer is able to be distributed in the depth or temperature ranges that the predators like, or other things like this? And it's very hard to get this information from, say, a single place. For example, you can do lots and lots of dives in the Monterey Canyon in the United States, which is probably where the world's greatest database of midwater animals has been put together. But there's only a certain number of combinations of all these different physicochemical parameters that occur there in Monterey Bay. And so there might be, say, pH extremes that they can't look at there, or the decreasing temperature always is low oxygen in that bay, whereas here off Australia it's different. And so by putting together data on these midwater animals distributions in various places around the globe in different water masses, we're able to get more of a handle on what are the factors that really affect the animals, rather than having all these close correlations between the different physicochemical parameters. Like temperature normally decreases with depth, so it's very hard to tell whether an animal likes increased depths or decreasing temperatures. So you need as many different combinations of the parameters as possible to tease out their habitat preferences. Oh, is that, what do we have here? Is that Colobonema, it looks like? That bluish tinge? Yes, that's Colobonema sericium, a repellent-ematid trachymedusa, hydrozoan, jellyfish. These guys, oh, you see the tentacles are of different lengths. It has a combination of long and short tentacles. When a predator tries to attack these guys, they shed some of their tentacles, and the tentacles glow in the dark. And while the predator is saying, oh, what's this? And going to check out the tentacles, the rest of the jellyfish swims away and goes and escapes. You can tell by the combination of lengths of tentacles how many times there's been a predation event or near predation event on the animal in the past. The Japanese name for that is the rainbow jellyfish, because if you shine a light on it from the side, the muscle tissue is very refringent. Ooh, that's a top down. Has a very, very thin stomach. Hanging down at least halfway down the bell there. And when you catch one of these animals in a net, normally they've lost all their tentacles. But by looking at them here in situ with the ROV, you can see that although the volume of the jellyfish may be only like a few cubic centimeters, the volume in which they're predating, if you look at the length of those tentacles and how they're deployed when the animal's undisturbed, you can kind of get an idea of the volume in which they're actually applying predation pressure on the animals around them. Yeah, you can see some of the refringent tissue and the rainbow-like colors produced by the lights there. Thank you very much.
Dhugal, just want to go back to bioluminescence. Well, as soon as it gets a little quiet, you mentioned the Siphonophore we were looking at earlier had this blue bioluminescence. What would be the purpose of a bioluminescence in a ctenophore?
So there's another Colobonema. There's a few hypotheses. Since it's very hard to observe like predator-prey relations, for example, in the midwater without disturbing the animals and like sit there and watch what happens with the bioluminescence, they're kind of educated guesses in some respects. But if you have an animal coming to chomp on the ctenophore, say an amphipod attaches, amphipods being smaller than the ctenophore, attaches to the surface and starts to like chomp away and eat it. The ctenophore will go, oh my goodness, and bioluminesce, and it's like a burglar alarm. And a fish might swim past and look at the ctenophore bioluminescing and seeing this spot that looks like a yummy shrimp or amphipod on the surface and say, oh, I'll have that, and pick that off and eat it without eating the ctenophore itself, in which case the ctenophore can regrow once it's no longer being chomped on. So that's one of the possibilities, that it's a burglar alarm kind of an effect. Without having eyes, it's kind of hard to see how they use it for communication. We do know that ctenophores, although they don't have eyes, if you shine a bright light on them, they stop swimming. Some species at least stop swimming. And you remove the light and they start swimming again. So it looks like they can sense light directly through the nerves, even without having those pigmented light sensors, such as eyes. Quite a few gelatinous targets around. That image there is showing us spiraling down into the canyon, yes? We're at 635 meters. Oh, that's another ctenophore.
What was the name of that jelly again, the Dhugal? The last one?
The last one, I think it's Ocyropsis, Ocyropsis. But I'll have to see the high quality video to be absolutely sure of that. Certainly looked like one. Quite a lot of these particles are moving. Oh, is that another Salmosis? Seeing a few more fish as well.
Brandon, the pilot's just saying that we're going to have to manoeuvre a little bit, get it more behind the ship. Currents are pulling us away from the Falkor, so a little bit of a manoeuvring going on here.
Okay. Roger that. Yeah, there are so many animals around that it's a good idea to maneuver back into a position where we'll be able to stop, observe, and sample an animal rather than get just a little bit of imagery before it flies on by, because we don't have the amount of tether we need to stop on it for any period of time. So I'm going to kind of half close. I won't close my eyes because there's still interesting stuff going past, but I'll try not to yell out to stop. There's still good video data-wise because the particles are still coming towards us. We're still getting in-focus shots for at least one frame for each particle. So this will all be analyzed later with copious cups of coffee, salps. It's like hyperspace in one of the Star Wars movies, doing the jump. Solitary salp zone. Actually, sometimes when you're going fast, you see animals you don't see when you're going slow. Going slow is really good for the jellies and the animals that don't move a lot, the sit and wait predators that are just kind of hanging around waiting for some prey to come close to them. Sometimes when you're going fast like this, you see other things like I've seen a swordfish before. You can see some big fish and squid sometimes when you come up them before they've had a chance to escape. Another salpzoid, solitary. So you never know what we'll see. Krill. Or at depths even where things like tuna could be diving to come down and feed on the midwater fish that are down here. So, yeah. Ooh. That looks like a Siphonophore. And it's still got its nectosome intact on the other side of the animal. It's very fuzzy, which suggests it's an apolemia perhaps. And if we are able to zoom in on the gelatinous swimming bells there and have a look at the nectar sac, the internal structure of the nectophores, and whether there are tentacles hanging down in between each nectophore or not, that'll tell us whether we have an apolemia. Oh, some of those guts are full and they're pigmented as well. It looks like he's been successful in its feeding. All those little tentilla are out there. Now, some siphonophores, the tentilla look like little copepods or shrimps, and they use them as lures to catch fish. Yes. Quite a small number of stomachs, really, for the length of the animal. Although each stomach has a single tentacle hanging out, and the number of tentacles there is very high. So I'd say there's a lot of stomachs there, maybe, that haven't eaten anything. Oh, that's nice. Cool. Thank you.
Sure is, do we have other cameras that help us position the camera, the vision you're seeing on YouTube and Facebook? And the answer is yes. We've got about 10 cameras all over the ROV at all different angles. It's almost like a full 360-degree view. But what you're seeing is the science camera. This is the main camera with we can zoom in and out, and it's the 4K vision. We also have a situation camera, which is much wider view. It isn't as high resolution, but it gives a very good wide view of everything that the science camera can see, and we can maneuver that around. So there's a whole range of different cameras that the pilots use here to try and center the vision on whatever it is we're looking at.
Yeah, the feeds from those cameras will also be very useful for telling what the animals are later, since we get to look at the animals from a variety of different angles.
Yeah, so there's two video types stored. There's the 4K science cam vision, which is what you're looking at the moment, and also the sit cam. So you get that wider perspective. And they're the two video types that we store for every ROV dive.
How are we going with respect to positioning with the ship now?
Yeah, we're looking pretty good now.
Right.
So temperature-wise, we're at 6.5 degrees C and we're at 724 meters depth.
Okay, and we'll be heading down into the Antarctic intermediate water. If we're not already starting to hit it, we'll be hitting it soon.
Yeah, that's right.
Correct?
That's right. Yeah, theoretically it's around 800 meters, but it'll be somewhere around 800 to 900. Beyond 900, most definitely will be in Antarctic intermediate water. We'll be down at temperatures 5 to 4 degrees Celsius.
One of the things we're wanting to look at is what's happening in the Antarctic intermediate water mass as it's moving northwards. It's been traced to about 4 degrees north off the coast of the Philippines, but there's only so far you can trace a water mass using the elements inside it or the characteristics such as salinity and oxygen or whatever. Oh, there's a Colobanina again. I can ID that so we can keep going. But yes, so the salinity, temperature, etc., gets diluted as these water masses mix. But the animals that are inside, you can't dilute them. So if we find animals that are indicative of the Antarctic intermediate water mass, and we can find them in water masses where we're not sure which water mass it is, we can kind of tell that it's Antarctic intermediate water. Wow, what do we have here? It looks like a Siphonophore. Oh, maybe this is the Euramphea, which is a lobate Siphonophore. We need to see the triangular projections that are now on the upper part of the animal and see if there are filaments, thread-like filaments projecting from them or not. That will tell us the genus. There's also an undescribed species of this family, which has black pigment on it, which lives in polar Antarctic waters. That looks like it's got filaments. So it looks like we have a Euramphea here. And it's very deep. It may not be the ubiquitous species that we find in surface waters around the world, including the Mediterranean. It could be a different animal, different species. So any video we collect of this guy may allow us to see some morphology that differs from the surface morph and possibly resurrect one of the many species that used to exist during 2 centuries ago, back when people were collecting the animals by hand and observing them and trying to find new species. Often there were very small differences, which they thought were species level differences, and there were a lot of species that were actually the same thing that were described as different species. But it's very, very hard to prove this because the Siphonophores don't survive preservation in formalin very well. So there are no type specimens to go back to, only the original drawings that were taken by the researchers at those times. Now with this new technology and the wonderful cameras, and being able to observe the animals in situ, we don't have to rely on 1 or 2 drawings by a scientist who may be using artistic license. We have this video record we'll be able to go back to define this as the image-based type specimen if it's a new species, or to redescribe a species if we can tell it apart from what's thought to be a single species in the genus. I guess we will find out when we go back and analyze this video. This is a great video, guys. All the different angles you need, yes. Very nice. And this one's got fully developed lobes, so it's more mature than the one we saw on one of the previous guys. Oh, what's that? Is that black pigment on one of the lobes there? Very nice. So in between those two triangular projections are the anal pores, and the mouth is in between those big fleshy lobes at the other end of the animal. So they're actually, the structure of them is kind of like a worm, with the mouth on one end and the anus on the other ends, which is different from regular jellyfish, where what comes in comes out the same way. They're in a different phylum. So although they're transparent and soft, they are not really closely related to each other at all. Thank you for that. That was an awesome video. Still krill around. I haven't noticed anything that I would say was a suggested shrimp. Which is kind of weird. Larvation house. Yeah, so many small targets around. Have to develop some kind of like a microscope camera to put on ROVs to image the smaller animals as well. One of the big problems with cameras, oh, what's that? Is that a lobate Siphonophore? Looks like it. Yeah, see if we can get a better image of this. Problems with cameras is depending on the optical setup, you're looking at like different volumes of water. If you want to see as many animals as possible, you can only really see the large animals. If you're trying to look at the very small animals, you're going to only see a few cubic centimeters at a time. So you really need an array of different cameras all in the water at the same time to be able to see the jellyfish from things that are like 0.5 millimeters all the way up to 40 meters long. Yeah, so it's a lobate Siphonophore. The tentacle bulbs, rather than being on either side of the mouth, they look to be up at the top of the stomach, which would make this a species of Bathysceroi. It's not actively swimming. If it was Bathysceroi fosteri, it's got nice big wide auricles. But if it was fosteri, it'd probably be swimming around, swimming away very actively. I'd suggest it's another species. I think there's still only 3 species of Bathysceroi described, although we do know there are others out there. And hopefully we'll be able to, with some images here, be able to see exactly what this one is. And if the images are good enough, describe it. Very exciting. Looks to be quite small. Bathysceroi is a very weird lobate. Just the structure of the internal canals, like the way the tentacles branch out, and the tentacle bulbs being at the top of the stomach rather than on either side of the mouth is different to every other lobate. We still don't know if they've evolved to look in this lobate form through convergent evolution, or whether they are actually close relatives of the other lobates. Oh, that's nice. Yeah, oh, cool. So it has a pigmented gut, so it's not Bathysceroi longilulia, which means the only other possibility for a described species would be Paragaster, which I think was described from New Zealand, which is much colder water, although we are in the water mass that's probably coming from up there. So yes, we'll look at the video of that later and see if that's Paragaster or if it's something new. Very cool. Reposition. I've got lots of exciting Siphonophores today. That's a Siphonophore. Only seen one, maybe two solmissus though, which is interesting because they're normally quite important predators of gelatinous animals in the midwater. And I think we're seeing fewer siphonophores than we saw during the last dive. Oh, we've got
jelly or siphonophore. Which one would you like us to follow? Both, obviously.
Siphonophore. I think the Medusa looks like a Helicreus minimum, maybe. Does it have like bumpy things on the outer side of the bell?
Yes, it does.
Okay, so that's Helicreus minimum. I can ID that. Let's look at the Siphonophore. So to ID it, we need to look at the nectophores, and we can see there it's a Pisonect Siphonophore, which means it has a series of bells. Ooh, wow. A series of bells and a float, in addition to the cyphosome. They're very large gelatinous bracts. Wow, what is this? It's not all fuzzy like an apolemia would be, although it's got a very long tail, very long cyphosome. That's very curious. Although it does look apolemia in that I can't see the nectar sacs. Don't. Oh, there goes one. It's dropped a nectar sac, a nectophore. Could we try and suck this up before he? They react to the light sometimes and throw away all their pieces, and it'll fall to pieces in front of your eyes. Wow, this will really fill the canister.
Oh, look how long this is. It just disappears into the gloom.
Yeah, it's probably an apolemia. There's a krill there too. If we can suck up the front portion of the animal, that'd be really great. Don't think I've ever seen an apolemia that has such well-developed bracts, like the gelatinous zooids that protect the reproductive organs and stomachs. Here seem very, very large, directly below the nectosome. Oh, nice. Oh, we're eating ramen. Look at that. That should be enough. We don't want to damage the rest of the Siphonophore that's in the jar. It should be fine.
You're out to laugh. You in Japan eating ramen, I'm sure you're used to that. It's just like that, isn't it?
Yes. Although if you actually ingested that, some of them have some pretty powerful stinging cells. So you might not be doing very much good to your stomach. Apolemia is one of the few Siphonophores where I've stung myself fairly badly trying to remove it from the sample canister. So please be careful that you don't actually put your hands in the salt water without having a plastic glove on when you're dealing with that guy, just in case he's one of those that do sting.
Yeah, copy that.
Jellyfish have whether they are deadly or not really depends on a combination of different factors. One is the stinging cell itself, the nematocyst, whether the the harpoon-like portion of it is strong enough to penetrate human skin or not. There are all sorts of nematocysts, which include ones that entangle crustaceans like get wrapped around the spines rather than penetrate the carapace. There are other ones that are very penetrative, and there's a range of different toxins in them as well. And you have different stinging cells in different areas of the jellyfish's body. You'll have ones for defense that work against fish, for example, on the X umbrella. You'll have different ones on the tentacles to help them catch prey. And depending on what they eat, the kind of stinging cell will be different. In Solmissus, for example, which is a narcomedusa that eats other gelatinous plankton, its stinging cell isn't very penetrative. It doesn't have to go through a hard cuticular chitinous outer shell like a crustacean has. It just penetrates jelly. So the very first spine is not very strong. But then after that, it has a very, very long rope-like, thread-like extension to it with barbs all around it. Is this a salp? Yeah, it looks like a salp in the foreground and an oikoplurid in the background, maybe. And so this thread with many barbs on it penetrates very deep into the jellyfish. And so then when the jellyfish tries to get away, at least some portion of that thread will be caught somewhere in the jellyfish's body. Other jellyfish have very, very strong spines, and they pop out with like the barbs on a fish hook to stop it from being pulled out of the carapace of the crustacean. So they've got different types around the mouth, which is used for handling the prey, different types to catch the prey on the tentacle, different types for defense on the outer side of the bell, and the toxins in them can be different as well. So you have a range of potential medicines because these are neurotoxins and other biologically active compounds inside each jellyfish. And there's an incredible diversity of jellies, thousands of species. Each could potentially have multiple different bioactive compounds in there. And at least here, we're able to get information on which species of jellyfish are at which depth. And although we can't catch large numbers of the animals with the ROV, the data that we get here could allow people to come out with nets and to fish at the depths at which we find each of the species. The DNA barcodes that we're getting will allow them to identify which species they are. And if they can get enough tissue to do protein extractions, or even working with the DNA directly more and more nowadays, they may be able to isolate these bioactive compounds and develop new medicines.
Hey Dougal, just for info, we've definitely pushed into that Antarctic intermediate water. It's 6.4 degrees C, but down at the depth of the ROV, there's a northerly going current. So the ROV is now facing southward, looking into that current that's sweeping northwards along the deeper Great Barrier Reef. So we've just reoriented the ROV, so we're now looking southward.
Roger that. That's nice because the particles will be coming into the camera and we won't be moving too far with respect to the seafloor, so we'll land on the right place for your benthic transects, correct?
Yeah, that's right. We're directly over the top of a big, deep submarine canyon, and we're aiming for the channel, the very channel floor, after we finish with the plankton audio.
Roger that. So, what will our next discovery be?
So you're talking about toxins for various jellyfish before. Of course, here in North Queensland, we have the box jellyfish. It's supposed to be one of the deadliest animals on Earth. Do you want to talk about the venom on box jelly there, Dougal?
Yes. So, my understanding, the box jellyfish, the real danger for them is the structure, the physical structure of the stinging cells are very penetrative to human skin. So if you get stung by one, that venom is going inside you and it's doing its stuff. Actually, Aurelia, the moon jellyfish, which has very weak spines on the stinging cells also has a very toxic toxin on it. But because it can't be delivered to humans through the skin, people don't really know about it, and it hasn't really been investigated. Box jellyfish, we're finding there are more and more species or different taxa. Things that we thought were brothers and sisters are actually distant cousins in some cases. So there's a lot more diversity out there to be researched, because each of these animals, if they're living in fairly similar environments, maybe their ecological niches are through the kinds of food they eat. And so one toxin might work for a little fish, another one might work for shrimp, another one might work for something else. And by studying the toxins of the various box jellyfish, you'll get kind of a handle on what they're eating as well. So it's interesting research to be doing. I believe you're doing that at James Cook University, yes?
Yeah, that's right. Yeah, those guys, mainly a summertime phenomenon. So the box jelly only is found in the coastal waters, not... we're talking about the Coronex fleckeri, the really dangerous one. Of course, we have these cubomedusae out in the oceanic waters here, but the really, really deadly one that can kill you in a couple of minutes flat if you get a good amount of tentacles on you. They're really only in the coastal waters, and so swim in tropical North Queensland, you should swim within the nets, and these nets are designed to keep these jellyfish out.
The little irukandji jellies that sometimes occur out here. We haven't seen any box jellies in any of the video, and we've actually been looking at the video right from the surface down into the deep. Whoa, I think that was a shrimp. And of course, the animals are quite small, and there's a lot of ambient light around there, which decreases the contrast between the jelly and the background. So they can be hard to see. So we can't say they're not there, really. But we have not seen one yet, have we?
No, I don't think so. But Teresa, one of our scientists at James Cook Uni, she did her PhD looking at these irukandji-like cubomedusae out at Osprey Reef, which is where we're headed tomorrow. So we may have an opportunity to see them out there. Just doing an ROV crew changeover. We've got Adam and Ben on the controls, so they'll just get settled in and we'll be on our way again.
Roger that. That should be one of the, it could be one of the potential highlights of the Osprey ROV dive. Is, I believe it's hypothesized the irukandji jellies hang around at kind of deep, colder depths. And then on a certain lunar cycle, they turn out all at once in the surface waters. So if we're able to see with these high resolution cameras, the irukandjis and at what depth they're hanging out in, that would be a really big discovery. The thing with these midwater dives is there's so little known that a lot of science proposals are for hypothesis-driven research, where we put forward a hypothesis and then we go to see if we can prove it. It's very hard to make a hypothesis when the amount of data about some of these places is zero. So this kind of an expedition like the Schmidt Ocean Research Institute is enabling, where we're able to do some hypothesis-driven research and some exploratory research, and we're able to go and look at places that we know nothing about to enable us to form these hypotheses to go back again with the research proposals for using other ships and other equipment to do in-depth surveys.
Do you want to have a closer look at... Sorry, Dougal, do you want to have a closer look at this one?
No, that's a larvation. That's okay. Oicoplurid larvation. You can see that animal is the white spot in between the two lobes. They're another kettle of fish. They're very important in terms of carbon transport, but they're also extremely fragile. By the time you collect them and bring them to the surface, they will have self-digested. The digestive enzymes inside the animal will digest themselves in the hour or so it takes to come back to the surface. That's Colobonemus sericium, which we saw shallow. Wow, that's pretty deep. 812 meters. So it may not be depth restricted. It might only need that temperature range. Cool. Thank you. Yeah, so these kinds of expeditions where we can go out and look at places that haven't been looked at before and do things that are very hard to do on some other platforms, and do them with such a high quality of the data that we're collecting as well is really, really exciting, and allows us to put forward these new hypotheses because we actually got data points now. You can see the gonads on that Coloponema there are running almost the entire length of the radial canals. It's a mature individual ready to spawn. They may spawn at very deep depths.
So the larvae just go out into the water column, or do they attach to anything at all?
I would say they just go out into the water column, but no one's ever hung around on a jellyfish like this for the amount of time that it would need to find out. That's another thing we desperately need, is a robot that can track, like find an animal and just sit on it for 24 hours or 48 hours. Because with some of the research that's done on land, you just follow the animals around and you give each lion a different name, and you can say, all right, 60% of its time is spent sleeping, 30% doing this, 10% doing that. We have no information like that for most animals in the deep ocean. There's a very small amount using a technique called biologging, where they put tags on the animals which record the time and depth and temperature, etc. But for animals that you can't tag, like this Coloponema jellyfish we're looking at right now, apart from knowing that it lives here and that it's this size and that the gonads are this developed, or you might be able to see into its stomach to see what it's eating, but that's where our information stops. We have no idea how this guy spends his day. Does he migrate into the surface at night? Who knows? How often a year do they reproduce? Who knows? And so trying to model how these things are affecting the ecosystem is very difficult with that lack of data. Every data point we have that increases our knowledge about them is very important.
Yeah, I was always amazed to watch Jamie Seymour and his jellyfish group. They were able to put surgical glue and glue little GPS tags on tops of these box jellyfish and track them along the coast in between acoustic loggers. So, but we're talking a jellyfish that you could, it's quite large. You can hold in your hand, though you wouldn't want to, not unless it was gloved, because these are deadly jellyfish. But yeah, using glue to attach to a jellyfish I always found that quite amazing.
Yeah, it's the surgical glue that will stick even when it's wet. It's pretty amazing stuff. Box jellyfish, the jelly is much more hard than the deep water jellyfish. Like some of these ctenophores, for example, the comb jellies can be 98 to 99% water content. So you try and put a tag in that, and it'll fall straight through the jellyfish and sink to the seafloor probably. So we need a robot with stereo vision to see how close it is to the animal. That'll just sit on it and track it. Another larvation house. I believe some of those box jellies, I think it was Jamie's work, they sit on the seafloor and wait until they see a fish swimming past and then swim up into the water column and herd it up and catch it. Pretty amazing stuff for a jellyfish.
Yeah, that's right. They've got very primitive eyes, so you can definitely see some sort of change in light. Yeah, the Ferrari of the jellyfish world. So they're pretty clever. Dhugal, we're just talking about time and distance here. We're now at 830 metres. I know you like to get to 1200 metres on most of these dives, but it's 12 o'clock midday here, Australia, and we're looking about another half hour for plankton work. If we want to get to 1200 metres depth, we're going to have to push it down a little bit faster. What would you prefer? Are you just happy at this speed and we call it quits at 1230, whatever depth we get to, or do you want to really get that 1200 metre zone?
Let's do a compromise and only go to 1,000, and at whatever speed we can manage for that.
Copy that.
So Hydra Medusa. 1, 2, 3, 4, 5, 6. I think that's a Helicreus minimum as well. Does it have gelatinous projections coming out on the rim of the bell, I think? Okay, let's keep going down.
It's definitely getting chilly now. We're at 840 metres, 840 metres, and it's 5.5 degrees C.
I think it's about 34 degrees C right now here in Japan. And here I am. Isn't it amazing that I can be sitting here in Japan in the mountains and looking at jellyfish at 840 meters below the sea surface off the coast of Australia? What a world we live in.
Yeah, it is. It's pretty amazing. And just where the ship is at the moment, we're only about 6 kilometres away from the edge of the Great Barrier Reef. We can see Lizard Island, which is quite a famous research station. We can see the mountains of Australia behind it. Nice part of the world.
Is there any activity in terms of like dolphins or whales or turtles or anything up in the surface? Any like megafauna you've been seeing from the ship?
No, that's not to say there's not plenty there, but it's all on the Great Barrier Reef itself on the shelf. So we're only, like I say, only about 6 kilometres away from the reef, and there's lots of turtles in amongst them. We're just coming out of winter now, and in this part of Great Barrier Reef during winter we get the dwarf minke whales. So they've now just departed, probably only a month ago. If you were to come here in the winter months, you can actually go snorkeling or just sitting in the water with these dwarf minkies. They're very curious, and they'll come up to you, and it's a big tourism thing that you can do. But at the moment, just sitting out here in these deeper waters, we haven't seen any dolphins. Also, the humpbacks have departed for Antarctica. So we do get humpback whales as far out north as where we are now, but they're now transiting all the way back to their feeding grounds in Antarctica, ready for that summer time when the sea ice starts to melt.
Wow. Are we able to brighten the video just a little bit? My screen's a little bit dark to pick out the animals. Please. Okay. Looks like a fish. There are very small fish called cyclothone. Oh, that's nice, yeah. Or bristle mouths, which are only a couple of centimeters long. Muscle tissue is very watery. They can't swim very far or fast at a time, but they're the most abundant vertebrate on the planet. And they do have swim bladders. They sit mostly motionless unless you disturb them in the midwater, feeding on ostracods and copepods. And we've seen a few of them as we come close with the ROV trying to get away. But we haven't seen any of the big lanternfish aggregations that at certain times of the year get together in canyons off the Great Barrier Reef for spawning. Oh, is that a jelly? Or a Bathycordius house, perhaps? Looks to be a larvation house. Okay.
Yeah, Dougal, I was keen to see the sea lanternfish as well. So a colleague of mine, Adrian Flynn, did his PhD looking at them. Okay, got something coming into view now.
That's a Siphonophore, it looks like. Could be a fast-swimming one if it's Bugmania. Getting closer to see. Oh. Looks like it could be a Bugmania, but it could be something else. Can we suck this up? Be able to do that? Quite a long nectosome compared to the size of the Siphonophore. Maybe a Halostemma. There's a couple of species described off the Great Barrier Reef back in the mid-1800s. Be nice to see if it's that or not. These Siphonophores, when you catch them in a plankton net, you often catch multiple individuals of different species. There's a second animal going through, which makes my point. And when you catch them all in the one net, it's hard to tell which piece goes with which animal. And being able to capture a single animal in a canister and be sure that all the parts of that animal are from that animal and not from something else is very important for doing that taxonomy.
Jelly jigsaw.
Jelly jigsaw, yeah. It looks to be the same species as that other one. So you could catch this one instead. Oh, yes. Nice one. Yeehaw. So that's a Physonect Siphonophore. We'll make a guess it might be a Halostemma. And we won't know until we can look at it under the microscope. So because of the difficulty in identifying the animals, because there's a range of different zooids, multiple types, and usually the species descriptions only have a description of one type of zooid, usually the nectophores or swimming bells. The bits that you catch in a net may not be the bits you need to identify the animal, so research on them is not very advanced. And I'm trying to think of papers written on Siphonophores in Australian waters that have been published since 1859, and not very many are coming to mind. So this should be a very important sample. Quite a lot of small crustaceans around, it looks like. Good Siphonophore food. Is that another one? Looks to be a different species. Very developed cytosome. Only a few nectophores. Oh, it looks like an apolemia. We know there are multiple species of Apolemia out there that are still undescribed. Yeah, that's an Apolemia. We probably better not, we get good video of it, but we'd better not suck him up in case there are pieces of that last Siphonophore still in the hose that might get mixed up with this one. Next animal we catch should be like a Medusa or something that we can really tell is different. That's nice. Yeah.
So what's at the end there? What's the big white mass at the end there, Dougal?
So that's the end of the Siphonosome, where the most mature zooids are, because they bud off from the base of the swimming bells. It looks like it's eaten something. One of the stomachs there is black pigmented and very large. And I think I'd put a bet on that there's food, and all of the other zooids there, because they're separate polyps that are like almost separate organisms, although they're attached to each other. They've smelt the food and they're all going to try and ingest it at the same time. So even though the stomach, the gastrozoid, will digest the organism and share the nutrients with all the other zooids, the zooids are kind of acting on their own, wanting to be the one who does the digestion. Thank you. If we can make that 1,000 meters. I think we've got a good shot of the nectosome there, and we'll be able to count the number of nectosomal tentacles in between each of the nectophores, which is one of the good characters for telling different Apolemia species apart. Wow, there's so many animals around, it's hard to make a choice. The majority of midwater animals are quite small, like fingertip size. But every now and again, you come across a very large and long-lived animal, like that Apolemia Siphonophore. I believe the Falkor, the SuBastian in Western Australia found one that was like tens and tens, maybe even hundreds of meters long. And so that would be a very old individual that has not been disturbed by a predator trying to eat it, and has been able to keep itself intact and just grow and grow and grow and grow, reproducing its zooids asexually, clones of itself genetically. And a lot of the animals are actually quite small. In the photographs you see of deep sea fish sometimes, you're imagining like this huge fish with these humongous fangs, which is could come and eat you up. But the actual animals can be, you know, a centimeter or two in length. They just kind of zoom in and take a macro photograph of them because they look cool. But there was an anglerfish that I thought was, 10 centimeters or more in size, and I saw it once out of the window of a submersible as it swam past, and it was like this big. I couldn't believe it. I pay more attention now to the scale bars or the text describing how large the animals are. It's really hard with video sometimes because you're zooming in or zooming out. The animal can be close to the camera or far away, and it's kind of hard to get an idea of size unless you're the one doing the driving, and you can kind of see how it's reacting to how much thrust you're giving the ROV. And you can kind of get an idea of size through that and the way it moves relative to the particles. But when you're analyzing a video afterwards, it can be quite difficult. One reason why we're putting the stereo GoPros on the SuBastian to try and get an idea of sizes.
Yeah, I agree with you. Scale's a little tricky, especially with cameras. There's always a bit of warping at the edges, but we pay a lot of attention to the two laser beams when we're down on the seafloor, which are 10 centimeters apart.
There's another Fisonect Siphonophore here. Looks to be a different species to any that we've caught so far. We look at the nectophores from the side. It looks like a Bugmania, perhaps. we call the snake Siphonophore in Japanese, because they swim sinusoidally like this. Although this cyphosome is quite small. Bagmania amelina. That's nice. That's good. Yeah, it's a Bagmania. There's no the nectar sacs inside the nectaphores are not Y-shaped. You'd normally be able to see when you look at them from the side like that. If it was a Y-shaped nectar sac, you'd be able to see like a whitish circle inside the nectaphore. But these there's no tissue that's in the same line of sight, so it looks whiter than the rest of the tissue. All of the nectar sac all looks to be the same color, so it's a very simple nectar sac. And the tail is not all fuzzy like an apolemia, so it must be a Bagmania.
Dhugal, you mentioned that really big Siphonophore that they found in the Ningaloo Canyon Falkor expedition early this year. I'm curious, when you get these really long ones, and let's say they get broken up through wave action or predator behaviour, they able to bud off and make their whole new colony, or like do they have to? If one of the neck zooids broke off, could it make an entire new colony, or they have to have all the different parts to be able to survive?
So the apolemia, or the Bagmania, like we just saw there, the Physonex Siphonophores have two budding zones. There's one just below the float, the pneumatophore, and that buds off the nectaphores, or the swimming bells, or in the case of apolemia, the nectasomal tentacles as well. And there's a second budding zone just below the nectasome, which buds off all the tail, the cyphosome zooids, including the stomachs and the protective bracts and the reproductive gonozoids, etc., etc. So the part of the organism that has that budding zone on it can reproduce more of that kind of zooid. So, for example, if the animal was broken up, normally the internal tube that runs the whole length of the animal is very hard to break in half. It normally wouldn't break on the nectasome. So if it broke somewhere on the cyphosome, you would have the budding area for the cyphosome still attached to the swimming bells, and that could reproduce and produce more cyphosomal zooids until the animal was long again. The tail that's been cut off from the rest of the animal doesn't have a budding zone anymore. So they're left to fend for themselves. And because they can reproduce and they can eat, and the bracts are neutrally buoyant and can keep the animal afloat, these things can persist like ghost nets for fishing, persist in the midwater just as the Siphonophore tails, and be eating and living for months without being the entire animal. But they can reproduce larvae and reproduce more of those asexual colonies from that discarded tail. But because they don't have the swimming bells, if something comes to chomp on them, they can't escape. And in some cases, they are slightly negatively buoyant, so they'll keep sinking down until they hit the seafloor, and then everything will come and eat them because they're a big carrier of organic matter into the deep sea.
Thanks, Dhugal. Great answer.
915. So hopefully we'll get that last 85 meters. And I'm surprised we have only found one Solmissus on this dive. Very surprised. They're fairly large in terms of the regular size of a hydrozoan medusa. So they stand out at a distance. They're normally fairly, you'll notice them and you'll find them and look at them, but there must be very, very few in this area. And I will admit the numbers of salps have been less than off the Cairns Seamount, for example, where we saw all those Solmissus on the first dive. so there may be less food for them. They are known to eat siphonophores, and there's siphonophores here for them to eat. But they are few and far between. What do we have here? It's a big feeding net or marine snow particle, I think. Yeah, it's not a jelly. Got a couple of animals in there chomping on it. Keep on going. It's a really weird shape, isn't it?
Hey Dougal, it's Marty here. Can you hear me okay?
Yes, thank you.
Rob's just stepped out to get a bite of lunch, so we'll be joining you for the last 20 minutes or so of this dive.
Okay. Another Phycenex Siphonophore here. This looks like the species we sampled 2 animals ago. The possible halostemma. Ooh, nice. That's a good shot. I get to see the nectaphores from above. Man, I bet that's Cupellifera. I guess we'll find out soon. See all those bright orange spots are the gastrozoids, the stomachs, which each is equipped with a tentacle that it uses to fish for its prey. The Siphosome's tail is made up of structures called chlamydia, which are little groups that repeat themselves along the stem. And depending on the species, they're made up of different combinations of zooids. Normally, there's a gastrozoid, a stomach with a tentacle. There's some kind of reproductive zooids. In some animals, both male and female. In some animals, their colonies are either male or they are female, depending on what taxa they are. There's often palpons, which are zooids that look like long, fat tentacles that actually taste and feel. They're the sensory zooids there. And then there are bracts, which are leaf-like or like kind of rounded shield-like structures usually, which wrap themselves around the other zooids to protect them. So other animals don't come in and like pick off all the stomachs off the Siphonophore, and it can no longer catch its prey anymore. And it's known that there are animals that will hang around a Siphonophore and wait for it to catch something, ingest it, and then just pick off the stomach and eat it. Charastaid juveniles, for example, fish, will do that. Some bathylagids will do that as well. (940 meters depth).
And we've just dipped below 5 degrees. We're at 4.9.
Nice and cool. Oh, we have a couple of targets. The right is a sinker, it seems, and the middle is what? A sinker being what we call the discarded larvation houses that end up looking like hot air balloons as they sink out into the deep sea. Is this a ctenophore or a house? Looks like a house, I think. I can't see any glittering of comos. In my video, at least. Yeah, it looks like a discarded feeding filter. Okay, thank you. These things act like little oases of food in the midwater. It used to be thought that the midwater was a very homogeneous environment, just like, you know, it's just made of water, so there's no rocks to hide behind or like plants to grab hold of. But as we see the environment using these optical tools, we can see it's very heterogeneous. There's large particles, small particles, string-like particles, big fluffy round particles. And the animals, the plankton, are not just like floating around in water. They're attaching to particles, hiding behind particles, etc., etc. Looks like another Siphonophore here. Maybe it's the halostema, although the Siphonophore is long, it could be a Bugmania. Let's get in a bit closer. Swimming as if it's a Bugmania. It might be a Bugmania. I can't see any whitish structure inside the nectaphores. I'll certainly be able to tell once we look at the high quality video afterwards. Get a close in on the nectaphores. Would be great. Yeah, cool. There's a lot of them around.
Well, there's a question that's just come up on the YouTube chat. Hi, Jason in Michigan. Is there a possibility to see a deep staria jelly, or is it too shallow?
No, we could see a deep staria. There's big scyphozoan jellyfish. Usually whitish. One species is kind of reddish brownish as well. They're really cool. They have no tentacles. They just look like a great big plastic bag, but they've got the canals inside them are anastomosing. They're all joined together in a network, kind of like chicken wire, in order to get the nutrients into this massive gelatinous body. It's got to have this interbranching canal system that will be close enough to the tissue to supply nutrients to it. So it's all reticulated like that. And you could find them at these depths. I've seen Deep Staria enigmatica, I think it was, at like 1,050 off Japan. Having said that, we're in an area that hasn't been surveyed by ROVs, so we don't really know. That's a Bathycordius house with the inner filter still there. And we don't really know where we would expect to see them distributed here until we find one. But we'll certainly be keeping our eyes out for them. I think we've actually only seen one scyphozoan the entire time. I haven't seen any Atolla, even Periphylla. Very few scyphozoans here. It's almost all hydrozoans. So far, at least. Another discarded house. That's a house too. You can see all the large particles attached to the external surface. The animal was still in there. You can see its body like a tadpole's head. And they have a little tail inside there, which is moving like this and making a water current to suck in the small particles that they eat. Oh, is that a Ceravoma eel or something else? It looks like a fish facing upwards. Whoop, there he goes. Silver-sided. Looked like a Ceravoma, I think. Coming down to 960 meters now. The last 40 meters of our dive. Well, the last 40 meters of going nice and slowly so we can see everything. After that, we'll have to speed up a little to get to the seafloor in time to do the benthic transect. And then because of the extra speed of the camera moving through the water, we'll still be able to see the larger animals, oikoplurid house, such as a Siphonophore that we just saw then, but the very small animals we probably won't be able to make out. We've been collecting a data set from around the world from 0 to 1,000 meters depth, 1,200 when we're lucky, oikoplurid house. So we'll be able to compare the entire Midwater community from this area with others in the South and North Pacific, which should help us tease apart what are the environments that each of these species prefers. Wow, another oikoplurid. So these filter feeders that weren't in the surface layers where you would expect would be closer to all the particles are turning up here in the midwater. This is a cyclothone, a bristlemouth fish. See, the stomach area is all black. It eats bioluminescent prey, and it doesn't want that prey glowing when it's being digested because it'll give away the position of the bristlemouth fish. So it's got a very darkly pigmented stomach area. And they actually have bioluminescent organs on their underbelly, which emit light at the same frequency and same strength as the downwelling light, so that they can hide their shadows in a process called counter-illumination. So any fish or squid or whatever that are looking up to try and see their shadows against the downwelling light can't see them because they're making their shadows disappear by emitting light from their underbellies. This is a piece of detritus. With a string of gelatinous material pulled along behind it. Most of the mucoidal particles we're seeing here are made by larvaceans or planktonic organisms. Oh, is this a cockatoo squid? Yes, I think we've got a cockatoo squid here. A cranchid.
Did you say cockatoo squid?
With his arm.
Yeah. Aptly named.
He's probably got his arms held up above the eyeballs. This is so they're not in his way when he's looking down, searching for predators that might be coming to eat him. And also, if it's one of the species with photophores below the eyes, because the eyes have pigment, they make shadows against the downwelling light. The animal wants to make its eyes disappear, so it emits light from the bottom half of the eye pointing down, and then it holds the arms up above the eyes, and so they are also invisible because of the light emitted by the eyeballs. The arms are very muscular. They have lots of muscle tissue, which means they polarize the light as it passes through them, and there are animals that can see this polarized light and will come along and try and chomp on the squid. You can also see its digestive organ, which is inside the animal, which is hanging vertically. It's attached to a hinge or a gimbal, and if you turn this squid around, the angle of that digestive organ will stay the same. It'll always be pointing vertically because it makes the smallest cross-section and the smallest shadow when it's like that. Hey, some of these animals will do things like eject their bioluminescent ink into the body cavity so they glow like a light bulb. Other animals eject a glowing blob of ink that's the same size as the squid itself, and then they swim away and hope that the predator thinks the glowing ink is them. Cool. Yeah, you see how the angle of the squid is changing, but the The digestive organ on the hinge is always trying to stay vertical. And some of these animals have a little cavity inside them that they fill full of ammonia, because ammonia is lighter than water, the seawater they're around, and it helps them maintain buoyancy. The ammonia also tastes really, really bad. So if a predator has eaten one of these cranchids before, they're not likely to want to eat another.
So Dougal, shall we keep going and try and get to that 1,000-metre mark?
Yes, please. Come on, soul missus. Where are you?
It's right at 1,000.
Right at 1,000. A 32 tentacled one. I want one. It'll be like at 1,002 after we sped up. Is that a Siphonophore? My feed has gone pretty jumpy, so I'm reliant on you to to find things.
Okay, we're keeping our eyes peeled. At the moment, we just have the Star Wars galaxy.
Okay. I'm surprised we haven't seen any little Piobius worms, which are polychaete worms that, unlike normal polychaetes, these don't have segments. And they normally hang around at depths like this, waiting for large marine snow particles to sink by, and they latch onto them and eat them. Normally, you can see quite a few. I haven't seen any at all yet. It's quite interesting given the number of larvation houses we've been seeing. But the most common described species now is supposedly a Northern Hemisphere species, or though we did find it off Chile. They may not be here. In which case, what is filling its niche? $990 meters, 10 meters to go. Where are you, soul missus? A couple of things there. They're all larvation houses.
Possibly a Siphonophore in the background behind.
Siphonophore, yeah. That's a lobate. Get in close to him and have a look. Maybe a Bathyscerode. You make out the tentacle bulbs. Are they on either side of the mouth or up near the top of the stomach?
Can't quite see just yet at the view that I have. Sit here for a minute and see if we can zoom in a little bit.
Roger. Should be 2 bright white spots. And then the other thing to look for is the stomach pigmented black or red or not. Certainly looks Bathysaroi-like. The stomach is pigmented.
Yeah, it looks more reddish to me. Black.
Yeah. So it's a tentacle bulb. Yep, tentacle bulbs of the stomach. So it's a Bathysaroi. Probably the same species as we saw just before. So if we get good video of this, we'll have 2 individuals, and we'll be able to say if there was a slightly strange structure, that it's not a mutation, that both of them had the same structure, therefore it is a new species.
Fantastic. Well, maybe we'll just sit here for a little minute and see if we can get some good video from different angles on this guy.
Yeah, that'd be great. And then we can call it a day, I guess. Wow. Look at the cones on the oracles go. Ooh. Yeah, it's got a much larger body than Bathysaroi fosteri. The thing will be to see if this is Bathysaroi paragaster, which was recorded from, where was it, Wellington Harbour or somewhere in New Zealand, in cold subpolar waters. We're down here in the Antarctic intermediate water, which has been swept from the south northwards along the edge of the Great Barrier Reef. So there is a possibility that even though we're so far away from New Zealand, it's the same species that's been laterally transported from the south. Or it could be something completely new. Something which the 4K video from the SuBastian should allow us to find out. Sediped.
Yep, we're getting moved around a bit here, Dougal, so we might have just moved away from the spot.
Let's go down then. Thank you. Should have some good video there. And we also have images from the situational camera and hopefully the GoPro still as well. The seafloor we're heading for is about 1,500, was it?
Yes, that's right. Do you want to stay on the line with us while we descend to the bottom, Dougal?
I will do so, yes. Never know when we might go past a giant squid or something. Or even better, an unknown Siphonophore. You've been seeing a lot of, oh wow, that's a classified Siphonophore, I think. One of the large rockets. You've been seeing a lot of little benthopelagic medusae, I believe, little reddish jellies, and some ctenophores as well near the seafloor. Self, I think. A few more meters to go. Haven't seen any tomopterids, the swimming polychaetes either. $1,000. Okay, thank you.
Thank you very much, Dougal.
Speed up and to the bottom then.
Yep. We'll continue on our descent down towards the seafloor. We'll pick up our speed a little bit. As we get to the seafloor, we'll continue on our dive, heading up this canyon wall.
So many particles at 1,000 meters depth. It's a lot of food. The canyon we're diving in must be acting as a channel, not just at near bottom depths, but in midwater depths as well. Sometimes we see at depths where the bottom topography is like a ledge, like a flat area before it goes back down to a cliff, that the sediment from there can come way out laterally into the midwater. And you'll have turbidity spikes at various depths, which correlate to the depths of shelves on the bottom, out quite a distance from where those shelves are situated. I've even seen supposedly near seafloor animals like rat tails or swimming sea cucumbers thousands of meters above the bottom in areas where there's a steep slope and shelves. Fish. Oh, that was big. Maybe Bathycordius house. It'd be really cool to find is a Stygium medusa, because they occur in the polar areas and are swept north as well. They can be tens of meters in length, but not like the long, thin Siphonophores. They're big and bulky and brown, and they give birth to live babies So they actually become pregnant, holding these little pink babies in their gonadal cavities. Surprised by the small number of midwater shrimp. There's no big red jumpy crustaceans. And the number of fish, even the cyclothone, which normally you see quite a few of them, not really that many. But there's gelatinous things all over the place. The SuBastian is a free swimming ROV, isn't it? There's no tether management system sitting above you, I believe. Krill. No larvation houses or big ones anymore. Still expect to be able to pick out fighter if they were here, but they don't seem to be. Something big. That's a larvation house. They still have the animal inside, although it's very clogged. And another. Ooh. The Siphonophore. It's like a fighter with a larvation. Take it we're directly above the transect start point now, so we're heading directly vertically down. That angle's good. Certainly seem to be fewer things around. Though with the speed change, can't be 100% sure. You still see the big things, and they don't seem to be here. So we're still doing our descent. It's 1,140 meters. We're seeing the occasional larvation house, large feeding filters coming past our cameras. We're doing a descent facing into the water current so that we get particles coming as best we can towards the camera, which is limited by the fact that we're trying to get down to the bottom now that we've finished our 0 to 1,000 meter midwater oblique transect. But we're still trying to get data just in case something big comes past on our way to the seafloor. As we approach the seafloor, which I'm assuming is at about 1,500 meters, since that was the target depth, we should start to see animals increase in number. Oh, there was a Medusa. Increase in number as we get closer to the seafloor, because there's normally more food associated with the bottom, either in the form of resuspended sediments or various animals that float up or swim up from the seafloor so they don't get eaten by the other animals that are stuck on the seafloor, such as crabs. That normally happens about 100 meters above the bottom. Sometimes the effect can be seen a few hundred meters altitude above the bottom, right into the midwater. At depths like this off Japan, we would still be seeing dinner plate jellies, Solmissus, large scyphozoans such as Tiburonia or Puralia. There'd still be quite a large number of animals, but where we are right now, if there are any animals here, they must be fairly small and difficult to see at this kind of a speed. So definitely the biomass is less than we would be normally finding when I do surveys off Japan. Can't really compare it that well to other places in Australia because of the lack of ROV dives that have been performed here in midwater areas.
Dougal, there's just a question there on the YouTube chat asking, what are these white particles composed of? Are they just the dust on the surface?
So what we call marine snow, which is called that because it looks like you're out on a snowy night, is a range of different kinds of particles. There are like dust particles and like non-organic material in there as well. But a lot of it is things like the discarded mucus from jellyfish or fecal pellets, the poo of various animals that have eaten at the surface, and their fecal pellets are sinking out into the depths. There are fish scales, there are the molts of crustaceans after they've molted, empty exoskeletons. There's possibly coral mucus coming from the reef, the nearby reef, which might be coming here as well. Material that's like the houses of larvaceans, oikoplurid filter feeding webs, or pteropod filter feeding webs. Bits of dead animals sinking out as well. So most of it is organic rich. Usually with depth, the amount of useful organic matter gets less and less as you go down in depth, but the very large particles sink very fast. And about the 40th houses, for example, have been reported to sink 1,000 meters a day, which means all of the organic matter there is still labile and able to be used by the organisms that eat it. So these very large particles become proportionally more important in the deeper habitats on the planet as a source of organic matter to the benthos and to the midwater animals that live below. And as all these things are sinking, they're leaving behind them a trail, like a smell. So when you're going through the midwater at a certain depth, it's actually a minefield of all these little trails of smell. And the plankton are swimming around and going, no, not that one. Oh, not that one. Oh, that smells tasty. And they'll go down and follow that trail of smell down to the marine snow particle that produced it and chomp on it.
Thanks for that, Dougal. That's very descriptive.
there are actually copepods that have evolved to eat. The only thing they eat is the exoskeletons, like the molts of other crustaceans. And they've got well-developed eyes, and there are like Vibrio-related bacteria which bioluminesce, and they cover the surface of these exoskeletons, breaking down the chitin. And these copepods have evolved eyes that can see these point sources of bacterially produced light on the carapaces of these the exoskeletons, and they hone in on them, and you open up their stomachs and they're just full of molts. So how's that for specialization?
You've got some fans on the chat there, Dougal. There's one comment there that says, I could listen to Dougal explain things all day.
There are other copepods that have got like brush-like structures around the mouth, which we think are also sensors to various different kinds of smells or pheromones. Unfortunately, a lot of these animals, we don't get to see them alive. And so we have to infer from the structures of the different parts of the body. We dissect them, or we look for clues as to what they do. There's a copepod, Tetrarhabdus, I think it was, that is one of the few crustaceans that has venom, and it injects this. It's like a hypodermic syringe thing attached to one of its mouth parts, and the mouth part, which is kind of flat and scraping, has a hole in it, so this needle can go through it and pierce its food, and presumably paralyze it so it can be eaten. So they probably feed on fairly large animals. And we found by dissection and doing sections and electron microscopy, etc., that there are glands there that seem to be excreting something, but we still don't know what that is. That work was done by Shuhei Nishida, who was at the Ocean Research Institute in Tokyo. A lot of fairly small particles, it seems, in the water now. Could be sediment related. Wonder how much lateral transport we have out here. That looked like a squid with bright white eyeballs in the middle. Gelatinous body and gelatinous tentacles on either side. Gelatinous arms. So. Oikofluid larvation, double load inner house structure. Wonder if we'll be lucky enough to see a deep star here or not. I'd be happy with the Solmissus. Can't believe there aren't any here. We have this Solmissus morph that we caught in Antarctica, and we have the same morph that occurs off Japan. They're genetically pretty much exactly the same. And we've got no samples from in between Antarctica and Japan. So I was really hoping that we would find some of these morphs here off Australia to fill in a blank. Right now, we've caught some of the shallower living morphs, but not this this big purple deep living morph.
When you say deep living, Dougal, what depth had you sampled that one in Japan and Antarctica?
In Japan, I don't think I've ever seen it above 600 meters, and 800 to 1,200 kind of is a good range for it, maybe 700. Of course, the water column parameters are different here, so it could be deeper because it's warmer in the surface here. We don't know. That's one of the things we'd like to find out. In Antarctica, it was occurring even quite shallow, only a couple of 100 meters. A lot of deep sea animals occur much closer to the surface in Antarctica because the temperatures are colder, even if the water depth is shallower. Unfortunately, in Antarctica, we're using nets. So the closest you could get for depth was like somewhere between 200 and 500, or somewhere between 100 and 200. It's an oikofluid house. So we don't really know where they were. But morphologically, they're the same, and genetically, they're the same. And we've got this great big gap in the middle that really needs filling. I said they're purple, but they are the regular whitish color of regular solmices. But if you get them at the right angle in the lights, they have this purple sheen to them. Who knows why? Some of the relatives of Solmicis, Solmicis probably has direct development. The larvae are released into the water and develop there. But some of its relatives, the larvae are parasitic on other jellyfish. And so they attach to a jellyfish, normally to its stomach or somewhere where they can suck out the nutrients before the host jellyfish has used them all. And they attach themselves with siphons, almost like gelatinous needles that pierce into the gastric cavity. And they suck out the nutrients and use it for them to grow, gradually increasing in the number of tentacles. They start out looking like little worms, and they gradually develop so that they become more and more jelly-like until they are able to propel themselves. They can open and close their bells or their velums, and at which time the movement separates them from the host jellyfish and they can go their own way. It's possible they could even eat the host jellyfish if they got big enough, I guess. But it's one of those things we still only have a partial story on a lot of this midwater ecology, especially about behavior. (1,385 meters). I think I'm seeing a shrimp here and there now. And that might have been a Munopsid spider isopod. Which are really cool isopods, relatives of the pill bugs, or slaters as we call them in Australia. Woodlouse, maybe, I think is another name. All of their legs are roughly the same length. Pod being foot, iso being the same, the isopods. And these spider isopods, they have very, very long spindly legs, which have little hairs on the end of them. And the hairs act like the hairs on the legs of water striders, which keep them suspended on the surface of the water. These allow them to walk in the viscous salt water of the deep ocean where they live. And one scientist wrote a paper on the way that these spider isopods walk. Left first leg first, right third leg second, kind of thing, analyzing the gait. And it turns out they walk in exactly the same way as cockroaches. So he must have thought this up, like sitting in his kitchen one night watching a cockroach walk, and said, "Hey, I wonder how isopods work," and found out they're the same, which is kind of weird, but also pretty cool, because the cockroaches have evolved to move on land, and the isopods are a completely different group, which is walking in water in 3D space, but they have the same gait. The isopods are also a very understudied group, because they usually lose their legs when you catch them in a net, and the legs have important diagnostic features on them. So people like Karen Osborne at the Smithsonian, for example, been doing work trying to catch these animals intact to try and tease apart the taxonomy. Oh, there's a shrimp. Acanthophira, maybe? Or Pasiphaid. fairly flattened. We're diving right down
in that canyon.
middle of this huge undersea canyon. Still no data from the altimeter from the look of things, so we're still more than 100 meters above the bottom. Normally the range of them is only about 100 meters.
Quite cold down here now, Dougal. It's 3.2 degrees Celsius, 1,550 meters.
Should put a couple of beers in there.
Adam's nodding, Dougal. He thinks that's a very good idea.
So what is our altitude right now? Do we still have no reading?
Yeah, so it hasn't kicked in yet, so not at that 100 metre altitude yet. 1598 is our bottom depth here at the moment. There you go, it's just kicked in now. The altitude has just kicked in, 110.
So we're bouncing sound waves off the seafloor now to try and see how far away we are from it. Oh, what's that? Altimeters can be deceivious little beasts sometimes, especially if you're going up cliffs or very great slopes, or the ROV is angled a lot due to currents or whatnot. So,
and this canyon is very steep-sided and narrow, so that'll be interesting.
Yeah. You see how the values are jumping a lot. This is because even a small change in angle of the way the altimeter is pointing towards the seafloor can, at this height above it, can make a big difference in the depth reading that you get. Are we expecting to see mud when we hit the bottom, or we're going to be on rock?
We're expecting to see mud when we get to the bottom. And maybe some coarse mud and maybe some rocks on the side of the canyon wall, but so far the canyon walls have had a thick draping of mud over them, depending on what depth we're at. So see mud on the floor, but what we might see on the walls, we're not too sure.
Okay. The big jellies are few and far between right now. Still no swimming peaks.
50 metres off the seabed now. Temperature is 3 degrees Celsius and we're at 1,550 metres water depth. Be too much longer and we should have the seabed in sight, maybe a few more minutes. And all the scientists have just walked back into the control room.
Lunchtime is over. all the scientists just missed that giant squid we saw go past, did they?
Yeah, they missed it. They were having a planning meeting in a different room.
Oh, good. Ooh. It was an eel.
25 metres off the seabed now. Still getting colder. We're down to 2.8.
Yeah, that's a pretty rapid drop, isn't it? Over this kind of vertical distance at this depth. Looks like maybe an amphipod swimming around there, top right. Is it a krill? Different species to the shallow one.
$15 metres off the seafloor now. What we see when we get down there. A 30-tentacled purple solmissus.
One can only hope. In, I think off Somalia it was, they were doing towed cameras, and when they got to the seafloor, which was supposed to be flat mud, there were just mountains and mountains of dead
Seabed in sight.
The oxygen levels were so low that there was nothing there to digest them. Here we go, seafloor. Certainly looks like mud.
Look like a shrimp.
Swimming.
Well, we're at 1,600 metres. We've more or less come to the end of our descent. We'll probably take a moment to sit here and readjust. Adjust the lighting on the cameras. We'll have a look around. We'll take a core in the floor. We'll take a push core here in this sediment. Then we'll continue on with the rest of the dive as we survey up this canyon. I'm going to sign off now and hand back over to our Chief Scientist, Scott, to take us through the rest of the dive. Thanks, Dougal. Great chatting with you.
Thank you very much for that wonderful Midwater survey. I'll follow you on YouTube so as not to use your bandwidth so much now, and wish you a successful latter part of the dive.
Good afternoon, everybody. Scott Nichol back in the commentary box here on ROV Falkor. Thank you, Dhugal Lyndsay, for taking us through our descent through the pelagic zone. Fantastic. Got all those jellies waiting for us to sample, to process once we get them back on board. But now we're going to move towards the benthic focus for the rest of the dive for the next 3 hours or so. We'll just landed at the foot of the floor rather of this canyon system, just to paint the bigger picture. We're in canyon number one in the series along the Great Barrier Reef margin that sits offshore from Ribbon Reef 9 and 10. And this is a canyon which is connected directly to the continental shelf and the Great Barrier Reef lagoon area through an ancient river channel, in fact, which is very clearly mapped, incising right to the edge of the shelf and then connecting to this canyon. So we're about 4 or 5 k's offshore from that position and from the shelf edge. And what we're going to do now is just get ourselves organised to take 2 short push cores through the canyon floor sediment. This is part of our standard procedure we've been using all along during this voyage, by which we use to characterise the surface and the near surface seabed sediments that have been deposited in these canyon systems, with a particular interest in understanding where the sediment has come from. Because we have a couple of options here in terms of sediment raining out of the water column. All those fine particles that we've seen floating past during our dive down to 1,600 metres potentially all settle out into the seabed at some times, and over geological time, of course, we have a build-up of those particles to quite a thick sequence. But also there's sediment which can make its way down the canyon axis, particularly during events which we call turbidity currents, where there may be an avalanche, say on the side of the canyon, which we've seen evidence before in some of the other canyons we've been looking at, and that avalanche would then lead to a mass transport flow down the canyon axis. We're not quite at the mouth of this canyon, so we're really sampling the pathway, if you like, along which that sediment is moving. So there goes that first core, readily pushed into that soft sediment without any trouble at all. And then it becomes a case of whether we can retain it into the core barrel. And that looks pretty good. Yep, that's another great example of an intact full recovery core for us. So we'll just store it in that container until we bring it up back onto the ship at the end of the day. And we'll take a second core sample at that same site, all about replication and giving us enough sample volume to work with back in the labs. We use these cores to characterise the grains, not only their size or texture, the combination of sand through to fine silt and mud, because that tells us a lot about the energy conditions that have transported the sediment to this location. So it's about the texture and the size, but it's also about the composition of the sediment. One of the other reasons for working in this canyon today is that it's had some previous mapping and sampling undertaken in it by J.D. Webster from Sydney University and Rob Beeman, who've collected a series of longer, deeper cores further down the canyon, which went in several metres, and they gave a very clear record of numerous turbidity flows down this canyon axis. High energy underwater landslides effectively, transporting all sorts of coarse grained material right down the length of the canyon. So these short cores will add to that story, particularly in terms of understanding the composition of the sediment as it's made its way down the canyon axis. That's great. Yeah, thanks Adam. Top work. 10 out of 10 for that one. So, nice and quick. So now we're going to have a bit of a fly. We'll take off. We've just landed on the deepest part of the canyon at this point, so 1,610 metres. We're going to move to the north and east initially, which will take us uphill on the northern wall of the canyon, and that's be a short couple of 100-metre transect. And then we'll turn around and come back the other way. That allows us to keep better control of the SuBastian, taking it upslope, particularly in relation to the ship on top.
Help me out there.
How does that look? Where are we at?
Right at the bottom here.
Okay. Yeah, okay.
I'm about to load in a new.
Just having a quick chat amongst the control room here, just to get ourselves sorted out. Up we go.
I'm just getting a full set from USBO because it's so much deeper than we had.
Let's go find some rocks.
We're going to find some rocks, yeah, thanks Adam. That'd be great. One of the little success stories we've had during this voyage, this part of the voyage, leg 2, is that we've been finding intact in situ bedrock at the base of these canyons all along the Ribbon Reef canyon system so far, and that's part of our objective today. And this coming into view right now looks pretty good. Okay. This looks a bit different. Yeah, we'll look closely here. Yeah, we've got some epibenthos attached to this block. We'll just carefully drift in, and I think we'll do some biological photography and sampling first before we
More accurate.
kick up any mud.
Looks like a bit of debris.
It does.
Like a big pocket. We should bump it and see what it is.
So Jeremy Horowitz has just come in
We just want to see what this
to the
is.
commentary. So just have a look at this. Thanks, Marty.
Okay, it's a bamboo coral. Actually, we should wait for the dust to settle. This might be a good one to collect.
Okay, great.
So you can see the nodes of calcium carbonate and gargonin repeating. Bamboo corals have been traditionally collected because they provide insight into the history of climactic events in the past. They're slow growing, they live a long time. And sampling at different parts of the colony, you could figure out what the environment, what the climate was like at that given time when it was growing. Additionally, we're working on the taxonomy of the group.
Should be a manual enough, right?
Okay. There we are. Nice.
So it looks like it's mostly dead and overgrown. There seems to be a few polyps remaining. So it'd be enough for us to sample. We make sure we preserve those few remaining polyps. So let's grab it.
Like that. Thank you, Adam. Yep.
So grab the whole stock, right?
Yeah. It's not too big, is it?
No, it's maybe.
Yeah. Yeah, potentially some zoanthids, but it seems like in the middle of the colony, are those the remaining polyps of the bamboo coral? I see James, you're on. So I think what we're seeing near the base maybe are zoanthids, and in the middle of the colony are the bamboo polyps. Is that correct?
Yeah, okay.
That's okay.
Yeah, we're good. We're good. We'll box that one. That's rather slump, doesn't it?
Yeah, certainly overgrown at the tip of the colony. So what we're hoping is that there's some polyps from the original colony, the bamboo coral, which we can preserve for DNA for molecular analyses.
Box one's okay?
Bio box one?
Yeah, they're all empty.
Great.
We just got down here.
I'd hope so. Cool. That's a nice start. The bio side.
All right.
So we might,
Maybe zoom around a little bit more.
yeah, and we might have a, what tool can we use to sort of scrape this face?
It looks kind of like mud almost.
It does looks
Definitely
muddy.
use the scoop because it's kind of got like an angle. You can use the arm just to see if it's soft.
Yeah.
Yeah, I'll just try that while you're there.
A bit of pokeroo.
We fully expect this to be very, very soft.
Oh yeah.
No, that's that's soft sediment.
So what could make that?
So we're thinking this is a block, a large block or boulder which has slid down a slope onto the canyon floor.
So underwater landslide, and this is the debris block that slid down. So it's come from higher up.
And it's quite likely your sample, therefore, has come from a shallower depth.
It is dead.
That is
essentially
dead, half dead, yeah.
Could
Okay.
it have recently fallen down and just now is? I mean, yeah, we don't know.
Yeah.
We'll see. But it is hard enough for corals to grow on it.
Yeah, exactly.
You know.
Yeah. Okay, so that's.
Why don't we take a look?
We'll have a
around?
fly around just behind, see, because we did see some other harder rock in one of the other cameras a moment ago.
It's fascinating to see all the chase fossils and the burrowing. So
That's
these
right,
are so presumably, a much older piece of seafloor higher up, but soft sediment, and we'll be talking about all chase fossils and things. The burrows on the sea surface is really interesting to see them on a cross section. This piece of block that's broken away. Lots of holes and burrows in there.
Right, let's get our head around this. So this, another
Anemone.
anemone, is that? Yeah, okay.
Anemone in the background.
Yeah.
Maybe polychaetes.
Right.
Big anemones. Oh, look at this one coming into view.
Nice.
So in a paper that I wrote with J.D. Webster and Angel Pujubanabio from University of Grenada was that these blocks, these landslide blocks at the bottom of the Ribbon Reefs could actually form habitats for soft, cold water corals and cold water coral communities like these anemones. And I think we're seeing pretty good evidence here that you can have corals attached to them.
I guess if it's just hard enough.
It's hard enough to
It's stable.
Yeah. So this is clearly a breakaway face. So that block may have just slid from up here. It might have just a few meters.
Right. Want to zoom out to see?
Yeah.
Slide back. You're
Yeah,
gonna slide
yeah.
back. Just gonna give you a bit.
Yeah, cheers. That'd be great.
This is the edge of the canyon. So, the canyon channel axis is like a river valley. The river can downcut into the sediment and then eventually it becomes unstable and collapses. Wow, that is steep.
That's nice. So we're currently facing, going along that north
Yeah, if you look
to
at
east.
the imagery here, you see how there's that dark patch that runs
Yeah.
all the way to the north. It looks like this level runs like that.
Yeah, okay. Yeah.
West there too.
Okay.
You want me to come up and over, or just
Yeah, let's do that. Let's just climb up a bit. Continue to ascend. 1597. It's just flying up the face of this
Big overhang.
scarp that this block is.
Look at the
Nice.
anemones. It's interesting dark layer.
That's right. But there's some interesting layering in this outcrop.
Can we see with this
Yep, sure.
without just kind of zooming in to see?
This is a
how we have
corallimorph.
bamboo? Yeah.
And these are basket eel homes, right? So eels make this sort of mucus, looks like a plastic bag attached to the rock, and they use it for, I guess, sleeping in.
Yeah, I think we're going to come up to that point and then we'll go straight across.
It'd be really neat. Yeah, I couldn't see this. No, I've done photos of John this week.
Okay.
Oh, he's yes.
Yeah, you just
zoom out a
zoom
little
over, I'll
bit.
just porch in and you guys take a picture and then you guys can
Yeah.
have a quick gander on.
Okay. Thanks.
Yeah, zoom out. There you go.
There we go.
Let's just get stable. Yeah, this looks like a legitimate soft coral.
Wow.
It's full of polyps. Okay, are you stable?
I am.
Okay. Let's
Zoom in now.
Yeah.
Yes. Interesting.
Yeah, let's grab this.
It's part of the slope, I think, so the floor of the canyon is many layers of sediment that are built up through time. Each layer allows a bit of motivation and burrowing, and it looks like the face is given way.
Just try to focus.
Yeah.
It's soft.
Getting there. There we go. Let's do better than that, can't we?
But this is all the old burrowing, right? It's now exposed. Usually we're looking down on it until the burrowing, and now it's. But we actually went on a lot before the off this thin screen, some big anemones. So this is just sediment. Yeah, layers, thousands.
No, it's not. We're back on.
And the canyon axis is like a river, river entrance, the channel down to but eventually
Yeah, there it is. It's on all there anyways.
Okay.
Becomes the anthony. That looks good. Certainly not the coral, but
this is all
got sea pen.
This has never been exposed.
See the stem there.
Yes. Yeah, okay.
It's really fascinating.
It's all right. Let's do it.
Like
Is it
that.
okay to go back to 1A as well?
Yeah.
But you control.
Look how sharp that edge is.
Yeah.
That's quite amazing.
It's falling out.
Oh, sorry.
No, it's just moving forward. Then you can dive in. Okay.
Yes. So this is certainly a good representation of what this species is. It's certainly not zoanthids, which I'm telling you, they're getting to me. Whether it's a coral with its polyps or a dead coral with zoanthids on it, it's hard to tell. Luckily, we have a good team of experts. We've got James, and we have colleagues off the boat as well, helping us identify what we're looking at. Zoom.
Exactly. Yeah, so the layer upon layer builds up because we have this quite a lot of sediment down through there. And then, that forms a layer of chalk, things like chalk.
as obstacles, whatever biodegradation is happening on the surface, as it gets buried, it gets preserved.
Oh, there's another bamboo on top.
What do you channel the cuts?
Those dark horizons,
And eventually,
whether that's organic.
cuts is where it's unstable and collapses, and we get this nice sheer face. It's soft. It's chalk.
Yeah. That'd be really cool. It's like a little just to compare.
Nice. Can we just zoom in while you're holding it?
Yeah, go for it.
So looking at it now, we could probably see that it's a bamboo coral. We'll just focus. Am I in auto or manual now?
You're manual, but I can cycle it here again if it's not doing it for you. And I don't know. Sorry. Alright, go for that, Jeremy.
Oh, sorry.
Yep.
Let's see if
We might be too close. We're right up next to the camera there.
If I saw that on the Great Barrier Reef, it would not touch it.
All right. Okay.
I mean, you could see it's bamboo coral. Now it's in focus. I would probably zoom in a bit more. So you could see on the stem, it's got those different nodes. There you go. You can see that. It's pretty in focus. Okay, yeah, that'll do. That looks beautiful. So it's pretty easy to tell the difference between this and the last sample we collected, so we could put them in the same box.
Same container right here.
All right, thanks. I can see it's sucked in.
I saw a coralline morph
It's
hanging
an attractive
down.
one.
So it might be one. Yeah. Oh, yeah, look at how soft that is.
Yeah.
You want to have a look around? Mel?
Yeah, I'll have it. I'll just fly back out and cruise along this section a bit. Thank you. Clearly a lot of biotubation in this outcrop face we're looking at. Numerous burrows. And then we've got these darker browny orange horizons running through the outcrop as we move up the section. Potentially organic rich layers in between the less organic rich clay sands. Interesting.
If I was on the shallow Great Barrier Reef, I would probably see clownfish sitting in amongst these.
Right, okay.
Layers are cool.
Yeah, they're continuous. That's very
I'll just give you a little view of the walls.
Yeah, that's good.
Pretty good view there.
Whiz. One of these legends. One of these sort of redox.
Big coral sticking under deep there.
Yeah, probably another bamboo.
That's probably more likely iron oxide.
Yeah.
So
Hiatus?
ferromanganese oxide.
Hi, Kathy. Welcome.
You know, like any stable surface that starts staining is dark colour.
Jeremy. It's another bamboo.
So there's the inset of the track we're currently taking to see get our bearings, everybody. So we're clearly climbing up one of the side walls. It's the northern wall. So down up to 1590 metres now. This is a sheer face, pretty obvious. It's lost those bandings now. It's just more of a up it goes. Good thing we didn't come down this.
Oh,
Okay. Yeah. So that's
This is staining. This is like ferromanganese oxide staining.
yeah. It is on the surface.
Usually a harder surface, like a rock surface that's been exposed to the water for a long time. Some hexactinella. Yeah.
Should we break a bit of that off? I think we'll break a bit of that off. Yeah, everyone's nodding.
Jeremy's got a sponge.
I do, and sorry, Jeremy, I was distracted by the.
Oh, no, of course.
So we'll take a sponge. Yeah, do you want to have a zoom in and have an inspection of that, and then.
Okay. My thinking is this is harder core.
So we've spotted what looks like a glass sponge. So these glass sponges are made of pure silica. And our colleague Merrick Ekins is studying glass and coniferous sponges from the Coral Sea and the Great Barrier Reef, and we've been tasked with collecting for him. And this looks like a good candidate. This is a morphology of a glass sponge that we haven't yet collected. So we could check it off our list.
Right. That's good.
So if this was a more coarser grey material like solutions, this might represent a low stand area, you know, where the
If it's hard, we can find the later on. There'll probably be more around here.
and you get a pulse of siliceous material.
Let's test that.
You might even be able to break it with
Yeah. I think we can. Just it's probably going to be very loose though.
On a crowbar.
Yeah.
We're just going to come up a bit and we'll land over the ledge there.
Okay.
So at first, these sponges, when we get them out of the water, they're very heavy. They so these sclerites that hold a lot of water. So when you get it out of the water, it's filled with water weight. So you think it's a very heavy glass, but then a few hours later, when you go back to grab your specimen, it's as light as can be.
Okay.
It was very interesting. Yeah. So, yeah. When we were dredging the Coral Sea last year for corals and rocks, we collected these really, really big, dense glass sponges, and you couldn't even cut it. You know, we had very sharp knives. We were trying to edge through it, So it was so dense of these spicules. And the next day, it was a different coral. After the water evaporated, we were able to pick it up. But before, we thought it was like the trunk of a tree.
Nice. Okay, that's
Beautiful. We're going to white balance on the glass sponge because it's pure white. All right, beautiful. Do we have the lasers on?
Sure.
Set to scale. There we go. So it's maybe 10 centimeters across. Okay. We can grab it.
Interesting.
And then
snail or something.
Yeah, I think we've seen them before. Kind of in the mud.
So what tool will we use with this?
Could we twist it like it
I
looks
mean, the
like?
claw may be able to break it up, but I think it might just collapse.
Yeah, under at the bottom, they look like some kind of looser bits. We just need something that's just slightly falling off. I might be able to grab it.
They're not that down here.
Just swapping out our ROV pilots right now. Cody and Ben joining the action. Oh, it's Cody and Chris, excuse me.
What am I doing here?
Chris, we're going to get that sponge there. The glass sponge.
And you can put it in 1A.
Gotcha.
Nice. Filling up bio box at a time. Yeah, it looks like there's a shrimp inside the sponge. It's up to him if he wants to come along for the ride or if he wants to get off at this stop.
He's a middle of the I'm sorry. Do you really want to? No, I just wanted to ask you too.
You may want to, you can probably answer that question. Maybe.
Sedimentary. Yeah. Yeah, we have a good question from Karen, talking about this layer. We've just been talking about it. At the bottom of this canyon, we do know that we're getting a range of different sediments through here, just from some sediment cores that were taken in the 1990s. And when we look through them, we can get layers of quartz. This is riverine quartz, layered with calcareous, softer sediments. So basically, as you go back through time, as the sea level cycles lower, you tend to get riverine quartz sediments coming down into this canyon valley, and as the sea level rises, you get it becomes calcareous limestone, and so you get these alternating layers of quartz and a sort of a calcareous ooze. So what we think that we're looking at is possibly a layer of coarser grained riverine sediments, because the rivers would have dropped all their sediment really close to the head of where this canyon is now. Of course, it's underwater by about at the shelf break by about 70 metres, but when sea levels were lower, the rivers would have basically drained right at the very head of the canyon. So our hypothesis is that dark layer we're looking at is actually just a resistant coarse-grained quartz layer. So we're going to try and get a sample of it, and that'll help us understand if that's the case.
Whoa!
It's like a kid's balloon.
You want me to get it or fly?
I'd like to see you put it in the hole without your arm.
trying to slam that.
Yeah. 1A, remember.
Test your driving skills. Yeah.
You want me to do
Background, right? Get one of those games.
There it is again.
So it is a glass sponge, but it's actually not as rigid as you think. It's fairly,
That's pretty good catch.
but you may want to not squish it for too long. It will break.
Got it. Gently.
All right, remember we said 1A, right?
Yeah. Why pop it up?
Yeah. Oh.
Oh no. It was it's one of these.
Now 1B.
It's one thing.
All right, 1B, 1B.
We can move it to A in a minute.
Fantastic.
So we'll just come back to that site.
The sponges are for Merrick, is that right?
Sorry.
The sponges for Merrick.
Yeah. I mean, it's for me, and I gift it to Merrick.
He's on the receiving tab.
Yeah, if he's good.
Coming back down, there we go. Good. So it'd be nice just to try and break off a little section of this
What about
face
the one that's
here.
jutting
What
out?
about that? Whether the claw will, I reckon it can, but whether it'll stay together is the question.
That little ledge you got right there?
Yeah, that ledge.
Stow your arm.
Just stabilising, putting out the arm of the ROV to hold us steady.
No, not yet. Well, yeah. That was crazy.
Now this may well just disintegrate, but we'll see. Okay. What will happen? Yes. Wow. Okay.
We got some rock.
Well, that's nice. That's really
1B.
Thank you. We'll put that in the box.
2.
The sponges in there. pretty close to. Yeah. It's going to be pretty distinctive.
This is quartz grains which are
Pretty good piece.
I think so. Yeah, it's held together, obviously, which is a great start. And looking at that fresh face.
Do you want to put it in B on top of the sponge?
You want to be in how big?
No, whatever they say.
Yeah, that's fine. If you want to weight the sponge down so the sponge doesn't flow down.
Sure. Yeah, it'd be good to get the sponge out of that because it's being bent right now.
Oh, I got this. Just so you know, if the thing's floating, it's going to go right and.
Yeah, that's a risk.
It's not a challenge for experts like you guys. Nice.
Fly chicken.
Great. Okay, that's
the
the sponges now.
We're about to see. We'll see when it comes up. It'll be a bit different, probably.
That's good. That's great. We're happy with that, team.
Really good.
Really interesting. So that will allow us to test a little hypothesis here that that's a coarser grained land-derived layer of river sediment, which has come down the canyon in the distant past when sea level was lower, and we had a direct connection from land rivers out to the edge of this canyon. And noting that there's more than one of these layers as we're flowing up this section, so this is a repeated process. Thank you, Cody. We'll keep flying up just along to waypoint 1. Okay, 1585 metres. We're continuing to ascend the northern wall of the canyon. As you can see, it just heading up above us into the distance. So we've lost that outcrop that's now below us, and we're now up on let's call it an undisturbed sediment surface. That all makes sense. Yeah. So we've taken a little step up. And so much more characteristic, intensely bioturbated, muddy sand, continuous cover. Essentially barren, in this view at least, of any epibenthos. Anything down here will be very sparse, as we've seen in all the other canyons at these depths, across these sediment surfaces, but still heavily bioturbated. So the life action is within the sediment. Burrowing crustacea, worms, and so forth. Here's one of our isolated epifauna coming into view. How we got, Jeremy? Got give it a moment to come into view.
Soft coral.
Soft coral. Yeah. Okay.
We collected something looking like this. Was it yesterday? The dives start to mend together after a while, but yeah. So it's certainly a
Anthomassus.
soft coral. Yeah, another anthomassus, right?
Yeah, so this is a Gorgon's head soft coral. It's an octocoral. Has these beautiful big polyps. Very obvious. We've seen lots of them, sometimes growing on rocks, boulders. You know, they have to attach to something usually, so you might even assume there's a boulder that it's attached to.
We're going to collect this and see what it's connected to.
They can be very colourful.
Right.
This area has never been above the surface. You know, we're down at 1,575 metres, so this has always been underwater. But the thing is, the head of the canyon has an ancient river channel attached to it. Of course, that is also underwater. It was probably exposed about 13,000 years ago. So the coastline ended at the of this canyon, but right here, this has always been underwater.
Beautiful.
Nice.
Gorgon's head, so it's like Medusa. Gorgon.
Is that the first, or you have
Sorry?
more video, or you have it taken?
I like this video. Hold on.
Looks like a facehugger. Facehugger.
A facehugger.
Alien.
Yeah.
Yeah, that's right.
Yeah, Kath McFadden says it's a strange colony shape. Yep, agreed. Typically you have this sort of round blob in the middle and the polyps grow out from it. But this is almost like a stalk.
Okay.
We'll do that.
Sorry, go ahead. Yeah, go to be and grab it. Yeah, so if you remember, 2 dives ago, we collected one that had that typical rounded shape.
Base, yeah, like the base of it.
And then
polyps.
had polyps around it.
Yeah.
And this one, they seem to be facing one direction, potentially current.
Oh, but I wonder.
Maybe the feeding is really in one direction and it's just
Okay.
grown, maybe.
Yeah, I was wondering that. Whether it's
Yeah,
like a tree in the wind.
right. Someone like Kathy would know.
Yeah.
What you say?
Oh, look how soft the sediment is.
What do we think it's attached to?
What's that?
What do we think it's attached to?
Nautilus.
Nautilus. Yeah. Been there, done that.
That goes down to
Yeah.
nothing cool.
Hopefully not a bottle.
Hopefully the treasure chest.
Yeah, that's right.
But whoever discovers a treasure owns the treasure, is that right?
Finders keepers.
So
Yeah, it's the salvage.
Oh,
Right of
sorry.
salvage.
Yeah, oh, geez, that's one of
Wow,
those.
it's really attached, isn't it?
maybe it's a rock.
Could be. Weird location.
Actually, the last specimen we had, I don't think it was. Maybe it was attached to wood. Because when we brought it up, there was nothing attached to the bottom, just a big bulbous. No, it was like 2 days ago. It looked like
Oh,
this.
that had a really long
That is
Yeah,
not coming out easily.
Dragon's
Wow, look how soft that is.
Gorgon. Top of the giant.
Nice work.
I have to make sure there's nothing below it that I'm a mess. Just a second.
There's a little squat lobster in there.
Must be living in the coral.
Sorry.
Yeah. When I got the last sample, I had to wash it in salt water to get all the mud off of it. It's like dense clay.
Stand by. I just want to see that hanging down there.
So just talking about sea level change, the last ice age here, which was about 20,000 years ago, the sea level had dropped about 100 metres from where it presently is. So only 100 metres, but it means that the coastline was right up hard against the head of this canyon. Hence we think that any riverine sediments from that ancient river would have dropped their quartz and material straight into this canyon.
Where you want to put it?
2, please.
5x2?
I don't want to take up all the rock boxes.
No.
Is that the base?
Yeah, got us.
Yes. You want to take a look at it? Yeah, why not? So is this how it's...
Like a big root, isn't it?
Yeah, is it anchored into the ground this way?
Yeah, I would guess that it was deep down there rooted in.
I'll try to do a zoom focus. There it is. Autofocus is useful. Okay,
Okay.
in the box you go.
Thank you. Okay, we'll continue up towards waypoint 0. 1 to 0. Just exploring. Here come a couple of... Oh, one. Oh, shadow.
Big jellies.
Jelly. Where's Dhugal?
Yeah. There's the tentacle hanging out. Great long tentacle.
Tucked it away there.
It's a good size.
Yeah. 30 centimetres across.
It's got a curtain around it.
It's got a... something on it there, isn't it? Burrowing into it. Ugh. Okay. Get through our dust cloud and continue on up 1570 metres and going higher. Again, back to this continuous muddy sand.
Yeah, just move it to the northeast a little bit. Yeah, we'll do. Thank you.
Polychaete worm? Oh, jelly.
Because we can't find
So great inset there of the canyon in 3D as we mapped it just last night, in fact, prior to deploying the ROV at first light today. There we are, zooming into our cross section that we'll be assuming. So what we've done, you can see we landed on the canyon axis floor and are now climbing that that right hand side, which is the northern face.
Just take a quick zoom
Makes it
in.
much easier to control our depth as we go upslope rather than downslope.
Something coming in on the right hand side.
Yeah, there is actually. Yeah. What are we seeing here, Jeremy, on the floor, do you reckon?
Well,
A little.
there are some small corals that I'm missing, but I'm seeing little glimpses of.
Okay.
So it's interesting, different than the last few days where really nothing's been able to grow on the seafloor.
But now it's a little.
Yeah, maybe it's flatter, maybe a little bit harder, allowing things to settle and grow, and not maybe there's less accumulation of sediment.
Yeah, that would be plausible.
Can we stop here?
Yep.
So this is one of those little, I'm not sure what it is, I just figure I might as well look at it. If we settle down, it'll be right in front of us. Try not to lose it. It's really small and thin branch.
What are you gonna do?
I'm not sure.
I mean, like, what do you want us to look see?
Yeah.
Where was it?
It's just gone up.
To the
It's
left,
down
yeah.
Yeah, to the left and down.
There we go.
Oh,
See
there it is.
the
There it
white
is.
object?
between
Yeah,
the-
it's attached to that.
Very faint at this colour.
Throw a little thrust up, get it smooth for you. See enough?
Yeah, we got it.
Okay.
Could it be? It's so thin.
It's a little pteropod next to it.
Maybe it's a bamboo. I just want to focus on it to see. And the pteropod up below it. I can see the shadow better than I can see the colony. Let's try to manually focus on it. Am I in auto or manual?
You're in auto. Manual now.
I'm in manual now?
Yeah.
Okay. The axis looks see-through. You saw dandelion Dhugal? Yeah? Okay. Okay. All right. Just curious as to what it was.
Okay.
Okay, we can continue.
Fly along things.
Yeah. We could try to stay low to the ground and we'll look outward for other, otherwise cryptic.
Is that low enough right there?
I guess as low as you can go without stirring up the seabed.
Yeah, that's already there.
Yeah, that's good.
Here, please, Jeremy.
Sorry.
Here, please. Jeremy might have to come to a hour, so sorry. Wow, and try to plan the field.
I'll stay down where the light is hitting.
You're so from waypoint 0, we're gonna Midwater.
Take off and shoot at the other side.
Straight through. Okay.
Yeah.
Do you want me to go somewhat fast? Get over there?
You can. Yep. We'll zoom out so you don't get seasick.
After we want to.
Is anyone watching the Deep Sea Biology Society talks today? 3 days of research findings about deep sea biodiversity and
Okay.
for whoever's interested in for 6 months. So, yes.
Yep, coming up. And just as we come along, there's a sponge ahead, perhaps. Just get Jeremy's attention.
Oh, tulip sponge.
Tulip sponge, I'm hearing.
That's another glass sponge.
Okay.
We've collected
There's a rock outcrop ahead.
at least one of these, so we won't need to collect another now. And there are zoanthids on the stem, so those are not polyps of, it's a glass sponge, so what you're seeing are these zoanthids that attach to the stem and connect via their tissue. And it can be very deceiving if you look at them and you don't look at what's above it. You might think it's, some sort of an octocoral. But no, these are zoanthids. So we're not going to collect it,
Okay.
but it is nice to see. They're very beautiful.
For sure. Now, I actually do see a bit of rock outcrop ahead. We'll just quickly zoom over to this little outcrop. Thanks, Cody. We'll just check it out quickly. It's a rock.
That's a rock with some anemones.
Yeah, it's kind of lacking any context here. It looks more like this loose material we sampled before. Another block of that, and I'm not trusting that as in place. Okay. We'll continue it. We'll fly out and zoom over to the other side of the canyon wall. Thanks, Cody. That's cool, turning around like that. So we're going to do this pretty quickly, just to get over there. Yeah, we'll go a bit of blue water for a bit.
This is green.
Yeah, wipe out 3 would be great.
Where's your ROV? We're going to turn around and head back.
Sorry everyone, back on audio now. We just went silent for a bit while we were relocating Sebastian to the other side of the canyon, but we've seen some jellyfish fly by en route. Dhugal's having fun. So we are still here with our audio feed. Not far away now.
What's that? Wait.
That inset view at the moment of the bathymetry gives you great perspective looking up the canyon towards the shelf break in that red colour. So we've been mentioning the fact that this canyon is connected to the shelf, and there's an ancient river valley which feeds the head of the canyon through the tides today, but in the past that was a direct connection of fresh water and land-derived sediments into the canyon head. So we've already seen some evidence of that, we think, in some of the sediments we've been looking at on the canyon floor in this dive. Okay, bringing the canyon back into view right now. Oh, that's great. So, yeah. So we're just staring at the. This is the southern wall of the canyon, and really steep face as we expected this, based on our bathymetry data. But again, it's some of that soft sediment that we sampled lower down, not a hard rock outcrop at the moment. So we'll just gently ascend up this face. A lot of debris. Okay. It's coming up. Thanks, Cody. Let's drift up. You can see clear layering in the outcrop. But again, these are canyon sediments which have been re-incised into by that same canyon. Wow, okay, that's looking more like a gravel potentially. Yes, and we'll just pause there, thanks Cody, and just if we can just hover on this little section, and I'll try and zoom in and get a closer look at this. Looks like we've got a terms of the stratigraphy, the layering of these sediments. It's very nice.
Just an edge in our area right
Yeah, that's good. Yeah.
Okay, I'm going to come into it a little bit.
Okay.
Land out.
Okay.
That'll give us time to let the ship catch up.
Okay. Because
This looks like tea.
I'll get back to it.
so it's letting just settle the SuBastian down for a moment and give us time to. We're not running away from this, that's for sure.
Right.
Yeah, that's great, Katie. Cool, mate. Ruby. Okay. Okay. Zoom out a bit more for a moment. So there's clearly we're looking at a bed of coarse to very coarse sand extending right across the view at the moment, and it's got a very sharp contact, lower surface into very fine-grained silt and clay. It's probably thickness of maybe 20 centimetres on estimate. Now this is likely one of these high energy, what we call turbidity current flows, which charge down the canyon, typically related to slope failures further up in the canyon, and it's pushing all this coarse sediment down. Now when this occurred, we really don't know at this stage, but we were suggesting earlier that it may have occurred more frequently during periods of lower sea level when rivers were discharging more directly into the canyon head, supplying the sediment. So I think we might try and take a scrape of this sediment. What can we use to scrape?
We got the bag.
I've got the bag. Let's try the bag.
Yeah, canvas bag.
Yeah.
Yeah, Chris, it's over. Scrape it up.
Yeah, we'll try that. We don't need a lot. Don't need a bag full. Don't fill the bag. Just a handful would be enough.
We can try to do it for. Try. I don't know how hard it'll be. Yeah, how hard do you think it is?
I can want me to dig on it first with the jaw.
Yeah.
Even if it's sand, you can probably rotate it in or twist it.
Give it a little touch.
Give it a go nut and see.
We'll area that right there in the middle. This layer right there. Yeah.
Because if we can get a sample of this, that'll allow us to look at the composition and say with a bit more confidence what the source of this is. All part of the puzzle for piecing together the evolutionary history of this canyon system. Because it's the evolution and the way these canyons change over time that controls the habitat that we're also very fascinated in. How does this look?
I might be able to do a core to that.
We're not bothered about preserving the stratigraphy, just give it straight in everybody.
Okay.
Yeah.
Yeah, that's
Let's do
right.
a core. Alright.
I mean, these are rare opportunities to sit here at 1,563 metres below sea level, looking at a very clean face, which is preserving the geological history of this canyon, which we can now sample and better understand. And as I mentioned before, and Rob was talking about this too, there are deeper cores, much longer sediment cores, which have been collected from this same canyon further downstream, if you like, of this site. Within those same cores, there's evidence of turbidity currents, numerous. I think it was 10 to 20 turbidity current events. So it's not surprising, but it's fantastic to see Such a good outcrop. Hope Jodie's watching. So we're going to have a go at a push core because we've got a lot of mechanical strength with this, even though it's coarse grained. Now, because it's horizontal, we've got a fair chance of retaining it. He says, hopefully. Always a degree of experimentation in these situations, but
that might be better. Yeah. I'm getting some in there.
yeah, some is all we need. We don't need definitely don't need to push it all the way in, Chris.
Alright.
Yeah. If we sort of tip it upside down, is it?
How's that?
Yeah, we've got. That's fine. That's fine. Yeah, we'll work with that. It's really that coarse fraction we're interested in.
There you go. That's it. Nice.
We'll take that.
Shoulder down. Just trying to get the angle there. There you go. You're going in. Alright, where to go?
Thank you, Chris. That's a nice little sample to play with later. Okay, let's continue our.
Look at this.
Smoke yourself out.
It's really get the wheel on. Okay, we need to move up and start to get towards our 500 metre transect if we can.
Yeah, that one little bit was holding the whole thing together.
Alright, come on to it.
Up we go. Good stuff. So seeing again repeats of these coarser beds outcropping.
Online on the conference.
Oh, behind us.
Sorry, Jeremy.
It's behind us. Oh, it's just a joke.
Up we go. It's a lovely section. Interbedded, just repeated cycles of these coarse interbedded fine-grained sediments. There's a long history of this behaviour within this canyon, which is consistent with the core record we had from that earlier work from Jodie. Okay, coming up, 1550. So we seem to have come over the crest of that cliff face now. 1545. Cup sponge. And there's another short face coming up, I think. Yeah, okay. Right, now the seabed's flattening out. It's risen up that cliff face over the crest. Still pretty steep, climbing all the time, but that's cool. So this is matching what we saw on the other side of the canyon as we left the steeper, lower slopes, those cliff faces, up onto this continuous sediment cover, bioturbated, very occasional isolated corals, sea pens. So coming into on screen now, thanks, just showing the cross section of the canyon in 3D. So we're on the left hand side as you look at it, which is the southern wall, and we're just climbing up that yellow dot on that profile is where the SuBastian is right now at 1530 odd meters. And as you can see, we'll continue to climb this slope and then it'll flatten out again.
Yes. This is the top of Russell's head. Doesn't touch straight on what it was, but it kind of makes some touch on his head on the front.
Yeah, there's no doubt these maps help to get your bearings in these deep dives. Otherwise, it's really tricky to really understand and put into the big picture what we're looking at. So that's why we do our mapping with the multi-beam sonar on board the Falkor. Prior to these deployments, they clearly help us all, including you all online, understand where we're diving, but they're also really critical for a safety point of view in terms of planning the safe directions to dive, because we've got to think about the fact this is the ROV is tethered from the Falkor, so there's a line running back to the ship, and we're dealing quite often with very strong currents at these depths, which can be another challenge for the ROV drivers. So it's all about doing the great science, but also keeping things safe, so we can get everything back on board at the end of the day with not too many dramas.
Good to the right.
Got Jeremy in the background just saying ask us to pan right, please, which we'll do. There's our good fish. It's a little shark I'm hearing. Cool. Oh, yeah, okay. Right. What have we got, Jeremy?
I don't know. Let's take a look.
Let's have a look.
Could be another
Settle down.
bamboo. Looks like a bamboo. Oh, yeah. So we've collected probably 2 of these now. So we won't collect it again. But yeah, a branching bamboo coral. We've seen a lot of bamboo corals on this dive. Interesting, you see a lot of one thing and not of others. So you wonder why.
Yeah.
So it's an interesting question.
Alright. Build an auto real quick.
The crustaceans, someone living inside. Okay, we're good. Okay, thank
You
you.
want a small branch of it?
Been there, done that.
Oh, you're sounding flippant. That's good. Don't oversample, do we? Yeah.
Whole thing?
We'll continue on. Thanks, Cody.
Okay. You don't want to sample it?
No. You in the mood to sample something?
Pretty.
All right, we'll get something.
Later on.
Pretty. Is that a nice word? Arm to the house?
What's this? A sea pen?
I don't know.
You don't know. You can just fly over it.
You don't look at it?
Nah. Yeah.
What's on your test? German type?
Jeremy wants a black coral.
I mean, something other than a soft coral.
Yeah.
So yes, black coral. There's also some sponges that we're still looking for.
Okay. We'll keep our eye out.
That's that. Another soft coral.
Just giving a quick shout out hello to Diatom Girl. Alex and Steph say hi.
Is that an acorn worm, Scott? I just went over an acorn.
Did we?
Yeah. Rachel was going to
Ah, there's a
Could we just back up a bit, please, Cody?
Yes. Back one.
Alex has just spotted a potential target we've been looking for.
To the left.
Do you want to guide us, Alex? If you,
Yeah, it's just a little bit there. There it is.
we're looking for a spiral pattern on the seafloor, if it's what we think it is. Did you see a burrow?
This here.
Ah, okay. Go ahead again. That.
Not an acorn.
That's not a fish.
Oh, it's an acorn worm.
It's an acorn worm. Target. Okay.
Target
So
word.
if we can
Yep.
sample that.
So we'll sample that with the sucker into a tank.
They're hard. They are hard.
You can blame Rachel Preslowski for that.
Yeah, she'll be excited. She's watching. 15, 20 metres, bang on actually. Okay.
When we suck on things and they get even on extremely low.
I could try just to catch it in the tube and then just place it somewhere.
So what's an
It's
acorn
really
worm?
fragile.
Now, when it goes into a set box, it'll stay there, and nothing else can go on top of it. It'll just
Break up.
disintegrate. Yeah, they're very fragile, very very fragile.
We had instructions on how to preserve it, didn't we?
No,
Okay.
All right.
Do you want to explain what an acorn worm is?
I have no idea, to be honest.
They make
So
cool little-
really
I'm
cool
Googling it.
spirals.
Spiral circles.
They make spiral burrows, which I guess the backstory here a little bit is our colleague Rachel Preslowski at Geoscience Australia, one of our marine ecologists, she's written a blog on the cruise website about Lebenspuren and the traces that these organisms leave on the seafloor.
That's sand.
Yeah, so this is an acorn worm, and for some time we struggled to understand or just to define a case of an active acorn worm in a burrowing behaviour. And Rachel found one earlier in the year on a cruise we had in Western Australia on the Falkor at a very similar depth, 1,500 metres. In that case, it was lying next to its characteristic spiral-shaped burrow pattern on the seabed. And we'd seen these spirals on previous surveys using other video techniques, not ROV, and always wondered what made them. So now, for Rachel, this is your second sample of an acorn worm on the Australian margin at these depths. We've got one from the western margin and now from the northeast. So
There's a bunch of fill.
Apparently they smell like iron.
It is. What?
Some fun facts. Closest living relative of the echinoderms, and they secrete substances containing iodine, so they smell like hospitals.
I heard they're really scared of sea squirrels.
Right.
Oh boy.
Good one, right?
That's a good one.
They try to get in the bag.
Yeah.
So are you worried if it's swimming away? Does it swim or does it? Well,
Yeah. Can we do?
I could try getting in the bag. I think it'll look easier. But might be time this week.
Yep.
Try that.
Just gotta be careful when you put it in. You should probably put it in the back.
Yeah, I know.
Thanks, Emily. That's good to know. Rachel's might tune in.
So the sample last time was the first intact convoy ever collected on the Australian continent.
Okay.
So we'll see if we can replicate it.
So what do we do with it again?
It's ethanol
Ethanol? Under a bottle?
and Siphonophore.
Okay.
And then to the museum.
Yeah. Eventually the top line is made.
Yeah.
Oops, the bag's full now.
Super. Rubber bag. Okay. Nice. Alright. Yeah, I'm gonna try to grab some of his surrounding dirt as well. Yeah.
Alex, you can clean out the bag.
Who's going to put the hand in the bag?
Shotgun that, or he's going to clean it out.
My hands are only for xenophiles.
They tear like wet paper. Okay.
They're super fragile.
Super fragile, so let's see how this goes. Wow.
Yes, we're good. Alright.
You wonder whether it's...
Somewhere I should be able to get up. Down need us.
We're breaking it, haven't we?
Got him.
He's got his tail.
Gee, they're fragile.
there you go.
Wow.
Is that complete?
Yeah, it's in one piece. It may have been damaged, but it's... Well done.
Gravity, come on, work with me, buddy. Don't be mean.
Oh, that's tidy.
He's in there.
Yeah, great work, Chris.
I will try to get rid of that.
gosh, look at all the slides.
Oh,
Oh, yeah.
you can process this one out.
Oh. Rachel's your only picture.
Sounds like Alex is Albert.
Yeah, I've wondered about that. Yeah. Okay.
Alright. I don't know what that big streaming part is. I think that's the big security. Okay, that was kind of like a fear answer going on me.
You don't need that.
Yeah.
That's
When the sea squirrels come in. Would you like to put it in the very back?
yes. 3/4.
So he doesn't get damaged.
There was acting like it.
So when sea squirrels attack these things, that's what happens right there. File box 3 open, Chris. I'm gonna take a look at the silk of it just to see.
Hey, you read that? Is the jelly going in the pump?
Self-sampling.
First time.
Take me.
Alright. We had one of the headless chicken monsters. Me and I was flying there with co-piloting, and there's something in the power box that just kept getting out. So we're trying to fix it with with one of the arms.
Yeah.
Sea cucumber, just the headless chicken monster right in the bio box.
Nice.
Close the doors. All right, cool. Sampling it. I think it felt so weird. You go over. I call it angles.
Yeah.
What did you and I catch and then we couldn't close it? I found that. What it was.
Probably fish.
Nice.
Good work. So that's only the second ever sample of an acorn worm collected on the Australian margin. Okay, back to transect.
Yeah, about 80 meters before we start showing. So.
Thank you. Okay. 80 metres to 500 metres. And there's another acorn worm.
You don't want to try to get the suction sample.
No, that'll be right. Can't have three, Rachel.
First one's great.
But good to see a spiral pattern from these guys. That's the yeah. We haven't seen that here.
Basically, it's the same exact way. There's another one.
None of them are cooking.
What have we got here? Okay, no. Not a coconut.
The soft form?
Nah.
See them?
Yeah, soft coral, octocoral. Interesting. It's different. But we'll pass on this.
Pass that.
Different than that.
Continuing our ascent up the the southern wall of this canyon. 15-20 metres. Not far away from our 500 metre standard transect.
Coming up.
So is that on site?
Oops, yeah, clear.
Just curious. Right. It's my friend. Learned it.
Yeah. Okay. Nice. So I think
Great, RV. Just to let you know, we're going to begin at 25 meter transect, so it's slowly keep moving in the same direction.
Alright, sounds good.
Alright, ready?
Tell me how fast you want to go when I start going. If it's too fast, let me know and I'll slow down. I'm going to come up about 2 meters.
So for the next half hour or so, we're going to complete a 500 metre
And we're cruising.
transect along our profile here. This is where we fly at a constant height and speed above the seabed. We don't stop or try not to stop.
How's that for the camera?
And the reason we do this gives us a regular record of seabed photos, imagery at a constant scale, constant horizontal scale. That's important for the quantitative work we do with these images. We have Alex Post from Geoscience Australia sitting in the control room here using software called Squiddle. And as we fly along here, she is annotating what is observed on the seabed at this constant height and speed. So it builds up a very statistically robust set of observations and measurements, which we can turn into measurements of the density of occurrence, frequency of occurrence of all the biota that we see, and also characterising the seabed substrate as well as we go. So it'll be a case of just drifting steadily along like this. We have been known to stop just for exceptional observations and things we do really need to sample, but for the most part we try not to. And again, we're working on a pretty tight time schedule today.
Oh, Rachel's on.
And here's another acorn worm or two.
Rachel's on Facebook right now.
Hey, Rachel.
Hey, Rachel.
Come onto the Facebook feed. Welcome from chilly Canberra, I expect. Yeah, so we grabbed one for you, Rachel. Yes, the guys used the bag sampling method. We effectively dug under the acorn worm, collected a fair bit of mud with it, and tipped it into the bag. Pretty sure it's intact, but we'll see once we get it up.
We're going to make Alex process it because it has so much mucus.
And we've got your instructions on how to process it. So we'll do our very best.
Question about how many push cores we've taken, we can produce some part of it. Well, 3 if you count the wall.
Yeah.
But haven't seen the spiral traces, Rachel, of the acorn worms that we've seen before on other voyages.
Very good. Thank you. No, okay.
So we're on a 500 metre transect, everybody. Constant height, constant speed. Oh, right, maybe a different species of acorn because of the fact that it doesn't have a spiral trace. Is that what you're thinking? Those on YouTube, I'm doing a bit of a chat with Rachel Preslowski on the Facebook feed right now regarding the acorn worms.
Tell her she needs to record that song she was singing for us when we were doing the course. Okay. She would sing a song to us.
Okay. Cody's just saying that we need to hear the song you were singing on board during coring. We want to hear a recording of that, Rachel. I have no idea what the song is. So already this dive has given us new stuff. Every dive, every day, we're finding new things for science. This canyon started off with some really good sections of exposed, what we are pretty confident, set of turbidity current deposits on the edge of the canyon floor. And now we're hitting the acorn worm city. I mean, this is the first canyon of those that we've looked at on this survey that have come up with the acorn worms. So ask the question, why aren't they in all the others? Nice steady ascent as we pass 15-20 metres on our 500 metre standardised transect. We've managed to do one of these 500 metre transects on every one of our dives on this leg. So at the end of it, we'll have a really robust data set to do our inter-canyon and intra-canyon comparisons of the density of occurrence of all the seabed biota and how they relate to the habitat of the canyon form and geomorphology and sediment types. So really happy the way this is all sort of coming together. This is our last day in the canyons in this part of the leg. We'll have one more day in a canyon planned for early next week, but tomorrow we'll be moving across to spend 2 days on Osprey Reef. Very different setting to where we are right now. It's for us it'll be a 8-hour steam out to Osprey from here. Rat-tail fish or two. Getting the inset again of our canyon bathymetry map. There it is. So, right, that's a great view. So we've just crossed a pretty flat terracey feature of the canyon, and now we're going to start climbing a fairly steep ascent. So we'll keep our eye out for any more of these outcrops, which we saw earlier, but we are getting higher up into the canyon wall proper, away from that valley. So you get that sense of perspective now. We're looking more or less face on into this very steep slope of this part of the canyon as we climb up.
ship a little bit. It's coming at a different angle now.
Okay, just adjusting things.
Yeah. But I was, no, I'm not trying to say. I'm just going to do it on the fly. Looks like it's about to start flattening out again. Yeah, I'm just swing over. Scrape on the starboard side.
There we go. Haven't seen too many of these today, the holothurians, but they've been on every dive, but I haven't noticed quite as many in this particular part of this canyon. That's fine. They move around a lot, of course, those guys. I've seen quite a number in the water column. Hello.
Yeah, course from the region.
Uniform was the word we're going to use here. Continuous sediment blanket right across this canyon wall. A little bit of a contrast to the other canyons we'd been working in previously this week, where we had a lot more extensive gully avalanche flows evident. But actually, when you look at the multi-beam from this canyon, the similar features are further up canyon from here, those gully features. One of today's objectives was to try and target some of those very steep surfaces at the canyon floor with the intent of testing to see whether there was any bedrock outcrop as we had seen in other canyons. Didn't see that, but we did see those marvellous sections of multiple turbidity current deposits. So a win nonetheless. Looks like we've got Andy Carroll online, I think. Biotubation. Lebensböden. and it's acorn worm number 32. Steady as she goes, 1440 metres. We've risen a good 100 metres vertically on this run. This 500 metre run, horizontally.
Straight up,
Yeah, there's the inset again, thanks. And again, we've said this before on other dives, it's these steep, very steep angles of these sediment piles is being maintained because of the cohesive nature of these fine-grained sediments. Very muddy, very sticky. But from time to time they do eventually fail with local mass movements, gravity flows, which then feed these turbidity currents we've seen, or the deposits thereof. Yeah, you're right, Dougal. Lots of acorn worms, and Jason in Michigan noting that on other voyages, acorn worms have created circles of their droppings, as it were. Yeah, you're right. We haven't seen that here, and Rachel Breslowski on the Facebook side of things was maybe suggesting it's a different species, different behaviour. They have a slightly different form, so. Definitely a contrast. Yes, thank you, Rachel, for reminding me. Fecal casts is what we are calling these spiral patterns on the seabed from the acorn worms. I struggled for words a moment ago. Fecal casts. How much longer have we got on our transect, do you think?
Not that much longer.
Yeah, nearly there.
Probably about 150 meters.
Okay, another 100 metres to go on our 500 metre transect, which is bang on time. That's good.
Yeah. Plus, going to consider we covered quite a bit of distance. You don't want to yeah. Well, it's been a while, so protection on the seafloor cover is higher than 500. Even north. Okay. Oh wow. Angle's good. Angle. Okay, we're 460 right now. Here. Almost. Bigger change.
Yes, we will have a count of the acorn worms at the way into this. 186 or 187. Shrimp bowl.
Where are we going?
Look, another holothurian down there. Haven't. Okay, coming to the end of our 500 metre transect in about 10, 15 metres. We shall continue the profile upslope. We've got 500 metres is the standard distance we fly. Then we'll go another few hundred metres, a couple more waypoints for probably another half hour to an hour, and we'll really just explore, sample if there's anything unique that we haven't already collected would be what we're looking at this stage.
And that's 500 meters.
500 metres, call it. Okay.
Yep.
Cool.
I'm not lined out right here. Take a look at this acorn worm.
Yep, that's good. Okay. Yeah, thank you. Well, that's right. We'll take a push core here.
Dump out. What's that?
There we go, acorn worm. There, we're looking at there, everyone's saying. There. Righto, let's pull out the coring gear. How many push cores have we taken? Leanne's asking.
3 so far. 3 so far. Oh, all up in total for the whole trip? So the trip, the whole trip so far, we're taking 27. Push core number 31.
Okay. This will be our 31st push core of the voyage. We've taken them, at least on leg 2, we've been taking them in pairs at each site, just to give us a bit more material to work with. Not always full recovery either, so but in these fine grained sediments they're pretty good. So the end of this transect will be at around 1,300 metres by the time we get there. Not bad, not bad. It should be fine, yeah.
I wonder how deep it is.
Yeah, it has compressed a bit, Chris. But that's fine.
Get that wording. Yeah, it's just it's pretty tight to try to shove everything upside. Not quite where I need to be. So yeah, try to spin it in. Or I'll go way up between other ones. Okay. Yeah, and reach between the middle ones. Slide it over. More time anymore? Is that it? Hold on. So you just shoulder right and pitch.
It's a delicate manoeuvre right now with getting this core in. Just the way the ROV's parked at the moment, but they've done it. Good one. We'll take a second. Thanks.
The second one, you said?
Yes, please.
I'm going to start working. clear, sir.
Or 8. Number 3 is a 3. Yeah. I think that was 6, so it's less confusing.
Do that one.
3?
It's gonna be easier. Keep coming up with your elbow.
Alright. I think that last thing would be that small out of this country.
I'm smaller.
That was tiny, relatively speaking.
Taking our second core now, and it's sliding a bit, but it's interesting. Okay, that's not staying in there. One's sticky. Maybe have another push. Give it a wiggle, as they say. Break the contact at the base.
Actually, since it's already been done, I can go deeper. I'm not that deep.
No, it's not like
Well, it's skinny. Let's see what happens.
I'll see how this goes. It's looking all right.
See how this one pops up. Roll.
I think we're good. Yeah. We'll take that as
go straight down the middle.
a good sample.
Yep, elbow down.